Hi everyone, good morning. We are going to start in a few minutes, okay? We're just setting up everything here. So
Sua. Eu sim. Obrigado. Espera aí que eu ainda não abri. Tem que abrir a sua apresentação. Possibilidade.
Well, good morning. Hi, everyone, again. It's a pleasure here to start the second day of our event in System Biologists, SysBio Rio 2022. Thank you all for being here, attending. Now we're going to start with the Professor Francisco Lopes in his second day of the short course.
Você tem que habilitar. Você está com o rosto. Hi, good morning. We just need a few minutes because I'm have trouble to share my screen.
Stefanella is saying that the host disabled the sharing screen. Stefanella, good morning. Can you hear me? Okay. We are done here.
Okay, we are set up now. So remember that this is a mini course. So I'm just talking about the basic definitions. Today, I will talk to both, to biologists and to theoreticians. First of all, I will talk something that's really is not new for theoretician, but maybe this is new for biologists. As I said yesterday, the strategy to build ordinary differential equation models is done in two steps. In the first step, we studied about the biochemical process. Then, based on this learning, we then build the reaction network model. Then we use the law of max action to find the set of ordinary differential equations. As I said yesterday, this step from the reaction network model to the set of differential equation, this is hard to do, but the computer can do that for us. This step, that is build a reaction network model from the biochemical process, is very hard to do. But one thing is very important here is that the computer cannot do that for us. So if you are a biologist, you have a great advantage because you have the biological background. You have the knowledge necessary to build the reaction network model, because you don't need to know advanced mathematics to build a reaction network model. You need to know biology. You need to know about the biological process you are studying. Okay, so let's suppose that you already have your set of ordinary differential equations. Like here in this very simple example, we are studying the dimerization. Here we have one monomer that reacts with another monomer, giving rise to a dimer. Okay, and this reaction is reversible. So here are the differential equation. I will show you a more easy way, a very easy way to find this equation. So if you have the equations, then we know that if you are in a initial state, the system will evolve to a stationary state. So we can answer two questions here. One question is, if you know the stationary states, the first question you must answer is, what is the stationary state? If you know the stationary state, or after finding the stationary state, then you can answer the second question, that is, what is the route that the system will follow from the initial state to the stationary state? But it's very important to say that the most important question is to find the stationary state. Sometimes you solve your questions just knowing the stationary state. Sometimes you don't need to know the route between the initial state and the stationary state. So first of all, we find the stationary state if needed the time course route. So if you are looking for the stationary state, our set of differential equations becomes a set of algebraic equations, okay? Because in the stationary state, the concentrations do not change over time. Okay? This is 0. Okay? So now we have the equation for the stationary equation, but we must realize that both these equations are identical. If we multiply this equation by -1, we will get this equation. Then, in fact, we do not have two equations here. We have just one equation. And we will see how to deal with that. We have two variables and just one equation. So let's suppose that these are the values for the kinetic constants. And we know then that all stationary states lie on this curve. Okay? It means that for each concentration of A, there is one unique concentration of AA. It means that for each concentration of monomer, there is one unique concentration of the dimer. However, you see that, as we know from basic algebra, if you have one equation and two variables, we have an infinite number of solutions. Then we see that our problem is not solved yet. We need to find one additional restriction. It means one additional information. And this additional information is the conservation equation. If we include the conservation equation, now we can determine the solution of the system. Why this is the concentration? Why this is the conservation equation? We can see here that anytime that two molecules of A are consumed, we have the production of one molecule of the dimer. So we can easily, in this case, find the conservation equation. Okay, then if I assume that C is 3, that we determine it by the initial conditions, then we determine the solution at the stationary state. This is a very simple case, but it will be useful for the next step. So I'm showing you here the steps we use. We have the biochemical process, build the reaction network model, use the law of max action to find the ordinary differential equations. Then we determine the stationary state, the conservation equation to solve the model. But you can think that it works just for very simple models. The question here is, what about the huge model, like that one I showed yesterday? If our model is a realistic and more a complex model, the reaction network will be not so simple. It will be very hard to follow these steps. This is because we use alternative roads that we that is the vector representation of a reaction network system. As I said yesterday, I will just show you a flavor of that. We don't have time to get in details about that. But I must tell you that this vector representation of a reaction network model has two most important rules. It helps us to find the set of equations. It most important, it helps us to know if the system has bistability. We remember that I talked yesterday about the definition of bistability. So bistability. So if you are looking for bistability in your model, it's very hard to determine if your model has bistability or not. However, if you use the vector representation of a reaction network system, it will be, I would say, easy to determine if your model has bistability, if your model exhibits bistability or not. So I will just give you a flavor about the what we can do with vector representation of a reaction network model with academic example that exhibits bistability. So here we have the first reaction where molecule B is converted into molecule A, and here one molecule of A reacts with two molecules of B, giving rise to three molecules of B. This reaction is very hard to happen because we need that three molecules to collide, to find itself. It's very hard to happen in the reality. This is why I'm called this example as an academic example, just to show you how to deal with how the vector representation can help us. So the first step, in fact, the most important step is to build the stoichiometric matrix. All the information from your reaction network are here in the stoichiometric matrix. Here we refer to speech A in the reaction 1. Here we describe speech B in the reaction 1. So in the reaction 1, we have the production of one molecule of A. Then this is 1 because we generate one molecule of A, minus 0 because we do not consume in molecule of A. Here we see that we do not produce in molecule of B, but we consume one molecule of B. Here we refer to the second reaction. Here we consume one molecule of A and we produce 3 molecules of B, consume 2 molecules of B. Okay? Then we have here the stoichiometric matrix, but one very important step here is to determine the conservation matrix. If you have a huge model, it's very hard to find the conservation matrix. But using basic algebra, we can find this the reaction, the conservation equation using these two relations. The first one indicates that the conservation matrix must be orthogonal to the stoichiometric matrix. It's making a lot of sense because they have additional information. And here, if we project the conservation matrix over the concentrations, we find the conservation constant. So in this case, here we are just solving this relation to our case, then we can easily find the conservation equation. So now we have both. Now we have the differential equations and the conservation equation. And from the differential equation, we get the stationary state. So now we can see how this solution works. So we are using here two different values for C. If C is 0.5, the behavior of the system will lie along this straight line, and the system will have just one stationary state. However, if C is 0.9, the dynamics of the system will lie on this straight line, and we can see that the system will exhibit two stable stationary states and one unstable stationary state. So we can easily determine where the system will be able to exhibit biostability. So the idea here is just to show to biologists how to deal with these equations. And but everything I show here, we can, the computers can do for us. But I'm showing you now something that the computer cannot do for us, that to build a reaction network model. Then now I will talk about an applied example. So we have here a simplified scheme for the NF-kappa-B mammalian pathway. This pathway is involved in a lot of biological process like immune response, inflammation, apoptosis, and cancer. So just one simple explanation how this pathway works. The tumor necrosis factor receptor in the membrane, after being activated by an external signal, it then activates the E-kappa-B kinase. This E-kappa-B kinase then induces the phosphorylation and the ubiquitination of E-kappa B. If E-kappa B is phosphorylated, then it releases NF-kappa B for nuclear translocation. Why that? Because E-kappa B, it binds to NF-kappa B and avoids impair NF-kappa B of performing is regulatory, is gene regulatory activity. So if tumor necrosis factor receptor is activated in the cell membrane, it will induce the phosphorylation of E-kappa B, then E-kappa B will be ubiquitinated and degraded. After being degraded, it will release NF-kappa B to go to the nucleus and perform its regulatory activity. But this is very important that one of the genes that is target of NF-kappa B is in fact E-kappa B. It means that after NF-kappa B starts perform its regulatory activity, it will activate E-kappa B. That is, remember that E-kappa B is a repressor of NF-kappa B. It means that NF-kappa B has a negative feedback throughout E-kappa B. So I'm show a representation here. NF-kappa B activates E-kappa B. E-kappa B has a negative feedback over NF-kappa B. So this is what we know. Okay. Now let's see that we wanna build a reaction network model to simplify the cell signaling network. We can see here that biology is giving us a challenge that is in fact, is very challenging to simplify this cell signaling network. So the idea here is, if biology is giving us a challenge, let's ask biology to help us. So the way we can ask to biology how to simplify this cell signaling network is to look for a homologous cell signaling network that we can assume as a simplification of this network. Where we can find that? We can find that in Drosophila melanogaster. Drosophila melanogaster has a very similar signaling network where some of the proteins involved in the Drosophila pathway are homologous to choose that are involved in NF-kappa B mammalian pathway. So we can then assume based on biology, that the tall Drosophila Patchway is in fact a good simplification of NF-kappa B mammalian Patchway. So let's work with tall Drosophila Patchway. So I will work with this result that was published in 2017, where they find a new function for E-kappa B. So let's explain you. E-kappa B is the repressor of NF-kappa B, while cactus is the repressor of dorsal. It means that dorsal is the homologous of NF-kappa B, while cactus is the homologous of E-kappa B. Remember, E-kappa B repress NF-kappa B, while cactus repress dorsal. Okay? So, before explain you about the tall Drosophila Patchway, I must explain you how the dorsal nuclear gradient is generated. This is very similar to the NF-kappa B Patchway. Here we have membrane receptors. If these membrane receptors are activated, if the tall membrane receptor is activated, it will signal for cactus degradation. If cactus is degraded, it will release dorsal to perform its regulatory activity. Then, in this case, if cactus is degraded, it will release dorsal to be transported to the cell nuclei. So we can see here that we have a high concentration of dorsal in the nuclei. It means that tau receptor is very activated in this region. And here we have a gradient where the activation of tau is reduced. Then it generates a gradient, a reduced gradient of dorsal in the nucleus. So here is the measure for this gradient. Here is the ventral region where we have high concentration of dorsal into the nucleus. And here is the dorsal region, that is here, where we have low concentration of dorsal into the nucleus. So now the question is, what happens in a cactus mutant embryo? If you understood what I said, or if I said in a way sufficient for you to understand, you will say, Francisco, if cactus is mutated, then the concentration of dorsal into the nucleus will increase, of course, because cactus is a repressor of dorsal. Cactus impairs dorsal to be translocated to the nucleus. If we reduce the activity of cactus, then we we are going to see more dorsal into the nucleus, correct? Yes, this is what we saw. We can see here that this cactus mutant will have more dorsal inside the nuclei. Okay, this is good. But what we can see here, something that is really unexpected. In this cactus mutant, the concentration of dorsal in the nucleus in this region is reduced. It means that here, the absence of cactus is increasing the concentration of dorsal into the nucleus. It's correct. But what is happening here? We should not see it here, because if we reduce the activation of cactus, we should have more dorsal into the nucleus. But here in this region, we have low concentration of dorsal into the nucleus. So it means that cactus play a positive role for dorsal nuclear localization. Yes, it was a completely new result. So we have two questions here. One question was already solved in this paper. The question was, does cactus play a positive role for dorsal nuclear localization? Yes. Now, the second question is, what is the molecular mechanism for this positive role? If you are a biologist, maybe your first idea was, let's go to the bench, let's do an experiment. But as a theoretician, I propose to you that we need a model. We do not need more data. We need data, just this data. Of course, more data for other questions, okay? So now I will show you how, by simple steps, how to build the model. So here are the effects we are reproducing in the model. Here we have tau dependent dorsal nuclear localization. This is exactly what I already showed to you. If membrane receptor tau is activated, it will induce the degradation of a cactus. Here is tau dependent cactus degradation. If cactus is degraded, then it will release dorsal to go to the nucleus. But we are in this model also describe another steps. That is the following. If we have, so here, these dorsal dimers, it has the abilities to induce the synthesis of a cactus in a similar way as NF-kappa B induces the synthesis of E-kappa B. So dorsal induces the synthesis of a cactus, and this cactus can bind to dorsal and do what we are call here dorsal sequestration by cactus. So here we have two mechanisms. One mechanism is dorsal nuclear localization that comes from the activation of tau, and we also have here basic nuclear translocation that is independent of tau-dependent dorsal nuclear localization. So all these steps here are well described in all the model. The novelty here are these steps. These steps are new, and these are in the modeling, and these are the steps we are including in our model. So I have just 10 minutes, so I will not explain how to build this reaction from this cartoon. But I will show you, in fact, it's something simple to do that. So if you have the model, then we can find the model partial differential equations. Then I will jump some steps, and I will show you our results. So we can see here that the model show two concentrations of those. They show, the model show the concentration of those that comes from this tau-dependent dorsal nuclear localization, and also here and here is the concentration of dorsal that comes from the direct dorsal nuclear localization. So the advantage of the model is that the model allows us to separate the dorsal that goes to the nucleus throughout the tau signaling, and we can separate it from the dorsal that goes to the nucleus direct. So, as I am saying here, dorsal direct, the model distinguishes the contribution of both dorsal translocation groups. Dorsal direct nuclear localization that generates this uniform contribution here, and dorsal dependent, tau-dependent dorsal nuclear localization that generates this gradient. So the final dorsal gradient is the summation of both. We can see that just throughout the model. So what we can see here is the following. So now that we have the model, let's see how the model behaves in the cactus mutants. In the cactus mutants, we can see the following. In the cactus mutants, we have an increase in the concentration of dorsal that goes throughout the uniform contribution. Because if we have less cactus, if we reduce the concentration of cactus, if we reduce the activity of cactus, we are going to have more dorsal free. If we have more dorsal free, we will increase the amount of dorsal that goes direct to the nucleus. We will increase these steps from the signaling. So if we increase these steps, it means that we will increase this uniform contribution. If we increase this uniform contribution, then we will reduce the concentration of dorsal in this region. Okay, so now we can then the model help us to understand how cactus, how in the absence or in the low concentration of cactus, we have less dorsal in the nucleus in this region. So to finish, so we can then conclude that at high tau activation, it means on this region where the activation of tau is high, a cactus improve dorsal nuclear translocation by inducing its phosphorylation. Here we can see that if we reduce the concentration of cactus, we are going to have more phosphorylation. Then it will reduce the concentration of dorsal into the nucleus. And at low tau activation, a cactus prevents dorsal nuclear translocation. So the final result is that kappa B in Drosophila, kappa B, it means cactus, has a positive feedback over NF kappa B. It means that we can have one step more here. It means that in Drosophila, NF kappa B activates kappa B. Kappa B has a negative feedback over NF kappa B, but also it has a positive feedback in the activity of NF kappa B. So the final question is, does this dual effect can be observed in mammalians? So to answer this question, we need more data and more models. And this because this is an open question. So this is our group, and we need to include here just Villenia. I did not have time to include Villenia picture. So thank you very much, and I'm here to answer your questions. Acho que acabou, né?
Any questions? Okay. Yes, there's no questions. Thank you very much, Francisco, for your presentation.
Great, you're welcome. Now, what we have
we will resume in 30 minutes. That's it?
Yeah, we will resume in 30 minutes. See you there.
Hello, good morning. Can you hear me?
Yes. No. Yes. Hi, Rafael. How are you doing?
I'm fine. I'm fine. Thank you. And you? Good morning to you all.
For coming.
It's a
Good
pleasure.
morning,
Morning, Adriano.
Wait.
Do you want me to share my screen?
Yeah.
Okay. Just a second.
Great. So,
Can you see the screen? Is that okay?
yes, this is
Okay.
great.
Okay, thank you.
It's my pleasure to introduce Rafael Valencia. He is graduated in biomedicine. He has a master's degree in biological chemistry and the doctorate in biological chemistry, both from Universidade Federal do Rio de Janeiro. He was a postdoctoral fellow at the Institute of Medical Biochemistry, UFRJ, and at the Institute of Biophysics, Carlos Chagas Filho, at UFRJ, and at the Pacific Northwest Diabetes Research Institute in Seattle, in the United States, as a guest researcher. He was a professor at the Department of Microbiology, Immunology, and Parasitology at the Faculty of Medical Sciences, Universidade Estadual do Rio de Janeiro. He was a technical scientific advisor at the Immunology Laboratory of the Clinical Analysis Sector at the Hospital Universitário Pedro Ernesto from the Universidade Estadual do Rio de Janeiro. He is currently a professor at the Universidade Federal do Rio de Janeiro, campus Duque de Caxias. He works on the following topics: evaluation of the mechanisms involved in hormone regulation and the study of physiological growth of proteins from the ABC subfamily of transporter and their relationship with the development of drug resistance phenotype in leukemia and other tissues. So, thanks again for accepting our invitation. You have the stage, Rafael.
Okay, thank you, Francisco. I'd like first to thank the organizers for the invitation to present this talk. It's an honor to be here, and the talk is focused on the regulation of ABCB1 and ABCC1 membrane transporters during tumor progression. Yesterday, Dr. Eliana Abdelai gave a lecture about cancer, and now we know that cancer is a world health problem. In the last century, we faced a shift in the leading causes of death in the world, in developed countries, especially. In the beginning of last century, most of the cases of death were related to infectious diseases, so like pneumonia, diarrhea, tuberculosis, and so on. and cancer accounted only with about 4% of the total deaths in the USA here, but also in other developed countries, the amount was quite the same. And what we observed in the end of the last century was a dramatic shift where cancer now accounts as the second leading cause of death worldwide. Only below heart and vascular diseases. So we know that cancer incidence increases with age. As we grow old, the risk of acquiring a certain type of cancer increases in both male and female types of cancers. And as we know that the world population is growing old, so cancer now is a major challenge of public health. So what we are going to talk about today are some molecular characteristics that are exhibited by cancer cells. So what are those characteristics? Well, we had an article review. It's quite old, but it's still updated from Hanahan and Weinberg, showing several molecular alterations that are related to the onset of cancer or to the progression of cancer. We can mention immune system evasion, induction of inflammation, the ability of infinite proliferation. But today we are going to talk about two alterations that are the evasion of cell death, which is related to the development of the multi-drug resistance phenotype, and also the induction of metastasis in solid cancers, which is started by the epithelium-mesenchymal transition. So what is the multi-drug resistance phenotype? Well, the MDR phenotype is defined as a phenotype in which cancer cells are resistant to multiple structurally and functionally unrelated drugs. So these compounds are different in action and in structure. And the first description was a clinical observation where patients underwent a chemotherapy treatment with a drug, let's name drug 1. The tumor disappeared somehow. The tumor couldn't be detected, but after some time, they exhibited its recurrence. The tumor size grew, and when the doctors tried to use the same chemotherapy drug or others unrelated, the tumor displayed resistance and this kind of unrelated resistance, which is characterized by the multi-drug resistance. Well, we know that multi-drug resistance mechanisms are multifactorial. Many alterations could concur to the induction of the MDR phenotype. We can mention the increase of expression of anti-apoptotic molecules. I'm sorry, this is in Portuguese. I forgot to translate. But the main focus of my work are related to membrane transporters that act pumping the substrates, the chemotherapeutic drugs, outside the cells. So the connection between the MDR phenotype and ABC transporters is quite well established. And it was the first mechanism described for the MDR phenotype. So it refers to the overexpression of those membrane transporters that uses the energy of ATP to transport several substrates, including chemotherapeutic drugs. And amongst those transporters, those membrane transporters, the ABC ATP binding cassette superfamily of proteins, they are of great importance. Those proteins, to be a full transporter, able to transport drugs, they may display two transmembrane domains attached to two ATP domains, ATP binding domains. So that's why those proteins are named ATP binding cassettes. But they also can be present as a HAP transporters, which need to dimerize in order to fulfill its transporter capacity. Well, the ABC superfamily has about 2,000 proteins that are present in organisms since bacteria to humans. So, however, only 3 of those proteins, they have significant clinical importance in the context of cancer. I mean, in drug resistance. So they are the ABCB1 and ABCC1, which are the main focus of this work, and also ABCG2, which was previously named breast cancer-related protein. So following the characterization of those transporters, of those membrane transporters, there was a search for drugs able to modulate the ability of those transporters to pump drugs outside the cells. And nowadays we know that there are several pharmacological inhibitors available. For ABCB1, we have verapamil, cyclosporin A, and others. And for ABCC1, MK571, indomethacin, and also verapamil at higher concentrations. So the problem of working with those problems is that they present a broad substrate specificity. ABCB1 usually transports positively charged molecules, and ABCC1 usually negatively charged molecules or molecules associated with GSH, with the reduced glutathione. So considering we have molecules, pharmacological inhibitors of ABC transporters, why is still tumor treatment still ineffective nowadays? Well, in fact, when we associate inhibitors of those proteins with classical chemotherapy, like cisplatin or, well, whatever the drug, what several papers have shown is that patients they displayed high toxicity in several tissues. And it happens because although those proteins were described in the context of cancer, in cancer cells, in cancer cell lines, they are also expressed physiologically in several tissues. So ABCB1 is expressed normally in barriers, blood-brain barrier, and so on, and also in secretory tissues like gut, liver, kidney, and others. And ABCC1 has an ubiquitous expression. Several tissues have been tested, and all of them were positive for ABCC1 expression. So it poses a difficulty in order to use an ABC modulator to produce a better treatment for cancer. Well, the other characteristic that was also mentioned by Dr. Eliane Abdellah yesterday is the metastasis, which is the ability of a tumor or some cells, certain cells, to detach from the primary site, to enter into the circulation and colonize a distant site in the body. So when it happens, normally it makes the treatment more difficult because sometimes it's hard to detect the metastatic tumor, and also because most of them, they present resistance. They display the resistance to drugs. So the first step of metastasis is characterized by the epithelium-mesenchymal transition. This process is characterized by a progressive loss of cell polarity and cell-cell adhesion, and the progressive gain of a mesenchymal phenotype. So cells usually lose their polarity and they gain ability to invade and to migrate through tissues to break extracellular matrix and so on. and they start to explain some markers, let's say markers of mesenchymal cells like encadherin, fibronectin, twist, and snail, twist 1 and snail, amongst just others, okay? So EMT is induced by several stimuli, but physiologically we can mention hypoxia inducing factor, also Wnt pathway, but in many cases it's induced by TGF-beta, which is upregulated both in scar tissue, in fibrosis, and also during metastasis, during the beginning steps of metastasis. So the relationship between MDR phenotype and EMT, it's quite well established, where ABCB1 overexpression leads to a more aggressive and more invasive phenotype. I'm showing a work from 2011 where authors, they separated cells from uveal melanoma that present a low expression or no expression of ABCB1 and high expression of ABCB1, and they showed that ABCB1 cells, they induce a faster growing of the tumor when injected in another animal. The tumor volume also is much bigger, and also they induce a higher number of metastases. So there is a relationship between ABCB1 function and metastasis and the induction of this migration that, but we still don't know how it works yet. But however, concerning ABCC1, we don't have no information so far. So as I mentioned before, overexpression of ABC proteins during the tumorigenesis leads to a more aggressive phenotype, and usually metastatic tumors, they present a poor prognosis, so because of the induction of drug resistance. However, the role of ABC proteins during tumor progression are still poorly understood, especially during EMT. So the main objective of this talk is to evaluate the role of ABCB1 and ABCC1 proteins on the TGF-beta inducing EMT. So first, we used as a model the lung adenocarcinoma cell line A549, and EMT induction was performed by the incubation with TGF-beta for 48 hours in the presence of absence of ABCB1 inhibitors. And first, we aimed to evaluate whether our protocol was of EMT was inducing this process, and we observed that incubation with TGF-beta induced the increase of encadherin, fibronectin, and also snail or SNI1 in A549 cells, showing that these cells are undergoing this process, the epithelium mesenchymal transition. And further, we tested whether ABCB1 expression would be altered during TGF-beta induced EMT. And we observed that ABCB1 positive cells were increased by TGF-beta. We had only a small part of the population that gained the expression of ABCB1, but not the whole population of the cells. But in fact, however, this is significant. On the other hand, although ABCB1 inhibition during the TGF-beta induced EMT did not alter any markers, did not induce snail and encadherin of fibronectin, the sustained sustained inhibition of ABCB1 using several modulators induced EMT markers, as we can see here for the induction of snail with cyclosporin A, verapamil, trifluoroperazine, and fibronectin. So simply the inhibition of this protein induced the expression of mesenchymal markers in A549 cells. So we tested further whether the activity of ABCB1 would be altered in those cells, and we made a protocol using a specific substrate for ABCB1 in the presence of absence of ABCB1 modulators. So when you use the modulators, usually the cells retain the substrate, which is fluorescent, and their fluorescence is higher. The protocol, first of all, we have a loading where the fluorescent substrate enters into the cells. We have an extrusion time where cells are allowed to pump. If they have the ABC proteins, they can pump the substrate outside the cells, so the fluorescence will go down. And in the presence of the inhibitors, the fluorescence will be high because of the retention of the substrate. However, what we noticed is that the fluorescence was the same after the incubation with TGF-beta, indicating that the process induced, although induced a gain in ABCB1 expression, its activity was not altered. So the partial conclusions was that considering ABCB1 induction was not related to protein activities modifications, we next evaluate the role of ABCC1, which expression is ubiquitous in this process. And we observed, just like the ABCB1 expression, and also observed a substantial increase of ABCC1 expression during TGF-beta incubation. But the inhibition of ABCC1 during EMT decreases snail induction, although it's a partial decrease, but it denotes somehow a dysregulation of the process when ABCC1 is not functioning. On the other hand, just like observed for ABCB1, the inhibition of ABCC1 also induced the expression of EMT markers, of mesenchymal markers, which can be observed for snail expression and fibronectin expression, simply by the induction of an ABCC1 inhibitor. And we further aimed to analyze what would be the impact of the increase of ABCC1 expression in terms of ABCC1 activity. So we performed a similar experiment using a fluorescent substrate, but specific for ABCC1. And what we observed was, contrasting to what we observed for ABCB1, EMT induces a higher activity of ABCC1 after the incubation with TGF-beta for 48 hours. And we can see here, that's a representative histogram from flow cytometry. We can see in control, when we use the inhibitor, the fluorescence, it's smaller when compared to cells incubated with TGF-beta for 48 hours. So this shift to the right indicates that the cells in the presence of the inhibitor, that they present a higher activity for this protein. So when we evaluate in each experiment, we see that after the incubation with TGF-beta, all the experiments presented a higher activity. So it means that possibly ABCC1 has a major role when compared to ABCB1 during this first step of metastasis, at least in this cell line. So to conclude, ABCB1 is significantly induced during physiological EMT using TGF-beta. However, only a small population was displayed an induction of ABCB1. We observed also that the pharmacological inhibition of ABCB1 during EMT induced no changes in mesenchymal markers, as observed for snail, fibronectin, or vencadrine. On the other hand, as I mentioned, the simple inhibition of ABCB1 induced upregulation of both snail and fibronectin, possibly ABCB1 and snail are regulated by coincident cell pathways, as usually ABCB1 modulators are also induces our ABCB1 gene expression. So it's a hypothesis we have to test next. And more importantly is that though many authors implied that ABCB1 is upregulated during EMT by other stimuli, especially using chemotherapeutic drugs, ABCB1 activity was not altered in this physiological protocol of EMT using TGF-beta as stimulus. So possibly ABCB1 present no role on drug resistance during TGF-beta induced EMT. So concerning ABCC1, we also observed that ABCB1 was also significantly induced during physiological EMT. Pharmacological inhibition of ABCC1 during EMT induced a decrease in snail induction. Possibly we have a sort of EMT dysregulation when ABCC1 is not working during this process. On the other hand, as observed for ABCB1, the sustained inhibition of ABCC1 induced also upregulation of both snail and fibronectin. And along with protein expression, the EMT induced by TGF-beta also promoted a significant increase in ABCC1 activity, which may account for increased resistance to drugs or also cell stressors during metastasis. So I would like to thank the organizers for the invitation to be here, and also the working group, Professor Lúcia Previato, José Osvaldo Previato, Professor Leonardo Lima, and postdoctoral fellow Kelly Costa. So thank you very much for the opportunity to have this talk today.
Great, Rafael. Thank you very much for your talk.
Thank you.
Let's see if we have questions, please. Let's see in the chat.
Do you want me to stop sharing?
Yes.
Okay.
Okay, Rafael. In your final comments, you more or less pointed what I wanted to ask is that what are the next steps of this investigation? So,
well, the next steps are, in fact, we have to test whether those cells during EMT they are significantly more resistant to chemotherapeutic drugs, but it's hard to fulfill this issue because most drugs that are used nowadays, they are both substrates for ABCC1 and ABCB1. So I'll have to use a broad observation, only checking whether common chemotherapeutic drugs would impact this cell viability during EMT. We also have some preliminary results showing that A549 cells incubated with daunorubicin, which is commonly used still nowadays, they die much more during EMT. This is a phenomena called collateral sensitivity, where cells that have a high activity of those pumps they, especially the ABCC1, they pump so much, the pumping activity is so intense that they run out of GSH, of reduced glutathione. So using a drug that has one of their facets is the induction of reactive oxygen species. So if the cell has no GSH, they die much more because they have no antioxidant defense, let's say. So what we have observed is that those cells undergoing EMT induced by TGF-beta, they die more, although they have a higher activity of ABCC1. But it has been shown for other authors, this collateral sensitivity. But we still have only a few observations. I have to repeat the observations to see if that's true, but I do believe it's true. I do believe that's what's happening.
Okay, thank you very much, Rafael.
Thank you.
Great. We are run of time, so I'm happy that I will have the opportunity to talk in person to Rafael to make my questions to him directly.
Okay. Thank you. Thank you to you all.
Thank you, Rafael.
Thank you.
Yeah.
Bye-bye.
Great. Now, it's my pleasure. Let's see. Perfect. Now it's my pleasure to introduce Dr. Jin Yang. He got a bachelor degree in physics from the Jin University, a PhD in astrophysics from the University of Illinois, and he got a postdoctoral. He was a postdoctoral research associate at the University of Illinois. Nowadays, Jin Yang is director of Computational Biology, Institute of Chemistry, Biology, and Drug Discovery, and the adjunct professor in the Applied Mathematics Department at the SUNY Stony Brook University, the university that I had opportunity to stay for 5 years and to meet Dr. Jin Yang in person. The main focus of his research is on the study of the fundamental mechanisms of biomolecular fold and the recognition, especially protein fold and the protein DNA interactions. He is also interested in the study of the underlying principles of cellular network. He is also studying the classical and the quantum non-equilibrium statistics mechanisms from the landscape and flux perspectives. Another focus of his research is on the reaction dynamics in complex environments, and he is also interested in the study of single molecule reaction dynamics. Welcome, Dr. Jin Yang. You have the stage. Thank you.
Can you hear me?
We are listening to you, but not so.
Can you hear me? Can you hear me? Hello? Hello?
Yes, we can hear you.
Oh,
sorry,
you can hear.
yes.
Okay.
Yes,
you can see the
can
screen also, right?
Yeah, yes, you can see the
Okay,
screen.
great.
Thank you very much.
Well, thank you very much. And I want to really thank Francisco for giving me this opportunity to share with you our recent works. And so as you can see the title, I'm going to talk about some of our own approach to the biological systems, especially I realize this is a systems biology workshop or course. So I'm going to use this our approach to focus on these systems biology. So I realize this is a course and workshop, so I'm going to give some basics of essentially formulate our approach. So we term that as a landscape and flux approach to the non-equilibrium systems. And so the first part, I'm going to talk about some of the theoretical aspect, which is going to be forming the foundation for this approach. And then I'm going to discuss several biological applications and see what we can do with this approach. And if we have some time, we're going to talk about some of these experimental tests on this approach. So let's begin. I guess this whole view graph, basically, I want to say that despite the textbooks where people mainly focus on describing the many particle system as equilibrium systems, most of the systems, or the whole world around us, is in non-equilibrium. And so the equilibrium is abnormal. The non-equilibrium state is normal. So if it's the other way around, then our world is not very interesting, and we are all going to be dead if we are reaching the equilibrium. So as the fact that we are here, it says that we are, if you consider that as a norm, that means that means non-equilibrium is the norm. We are the norm, if you think that way. Anyway, so the systems typically people are studying, or it's relatively easy to study, is the closed system, no environment, such as we are showing here on the right. But in fact, most of the systems that we are studying cannot be isolated from the environment. We are not isolated from the environment. We have to eat and drink, right? And we have to exchange ideas with the others and the environment. So we're not completely isolated from the environment. So this kind of systems are everywhere. And the question is, this is the norm of the system, cannot be completely isolated from the environment. And these are the systems we should put more attention to. And this is also about the systems biology, because we go around and talk about the other things, but systems biology concerns about gene regulatory networks and all other things. But the most important aspect, at least from a physical perspective, is essentially this open system nature. Basically, a cell has to exchange information, energy, and materials from the outside, and to be able to survive and sustain its life. And that's the key. But although the equilibrium system is described quite rigorously and quite well established, such as statistical mechanics in physics or physical chemistry, the open system, such as system always interacting with the environment, is not quite well established. this is the part which leaves us the room to explore more, and it's that's the sort of the focus of our study. And here is just an example that a cell, really has lots of components, and if we simplify that, some kind of a gene regulatory network, for example, signaling molecules coming and activate the receptor proteins and pass this information to the gene, which is essentially through a signal transduction network, and eventually got to the gene and initiate some of the changes here, and eventually this this response to this input will be in the form of some kind of signals coming out or some of the changes in the cell. So the whole process is non-equilibrium. It's involved with many ATPs and all these processes. So so that's kind of things that we are interested in. And the physics or mathematics of describing such things as saying, for example, gene regulatory networks. Francisco just gave a very nice introduction about thinking about these networks in the cell as a chemical reaction soup. So you can imagine they are many different kinds of molecules, and they are crowded into the cellular environment, and they interact with each other, initiate the chemical reactions so that form a chemical reaction network. And this chemical reaction network in mathematics, you can write down the evolution equation. Essentially, X can be thought of in the systems biology context or gene regulatory context will be representing the concentrations of the molecules in the cell. And Fx is the driving force for this. And so this is a very generic type of equations called the nonlinear dynamic equations. And you can also think about this as X, sort of a Newton's second law, except that the X double dot we ignore here because here we assume that friction is very strong, so that the double dot term, time second derivative of the time can be ignored. This is very similar to the situation when you jump out of the plane from the sky, and instead of going down ever increasing speed, in fact, you quickly reach the terminal velocity, and that's the situation like here. And you actually hit the ground with the constant speed and not in the g, the acceleration. That's the real situation. But in any case, so you can understand this equation as the concentration change or the evolution is determined by the force. It's like a Newtonian force, like quantity, except this is the chemical reactions determined by that. The typical way of finding this, the features of this, is to find the fixed point. Fixed point means they don't move, and x dot equal to 0. And then do the linear stability analysis around this, means do some small changes and see what happened. And in this way, we can figure out what happens near this neighborhood of the fixed point. And then that gives you the local stability information. However, there's a drawback that I want to immediately point out, is this approach typically doesn't give you the global information. By global, I mean, if you have two fixed points, if I want to connect them together and ask what's the relationship between these two fixed points, the local stability analysis does not give you this information. So if we want to know this global information, this is very crucial from one fixed point to the other, because this is related to some, for example, in the cell or systems biology language, so this could represent the cell types. I mean, like different gene expression patterns, how you go from one to the other. And this is absolutely important information that we're going to study. So to give you a graphic sort of illustration, so I use the gene regulatory network as an example. So this is a three-dimensional illustration. So each axis represents one gene expression. I mean, suppose we have zillions of genes, but I only show you three different types of genes. So gene X, gene XA, XB, and XC. So a point in this high dimensional space represents a state in the gene expression profile or in gene expression state. And the trajectory represents how you go from one state to the other, the dynamics. Okay. So this is the genomes, and these are the genes, and they are forming interactions with each other, right? These are genes, and they form interactions with each other and form a gene regulatory network. And if you go to these the coordinates as I just mentioned, so going from one state represents a state which is formed by all these genes involved in the network. So this represents the expression level. And so this represents one state of the collection of the genes. Each gene has its own gene expression level. So this represents one state in the gene expression state space. And this is a different combination where this gene becomes higher in the expression, and this becomes lower, this becomes lower. So the next moment or next few moments, it becomes a collection of the gene expression pattern becomes this. Certainly these two are different, representing two different states. So, and this is what happened when you look at from the gene regulatory network perspective, the system or the cell goes from one state to the other in the gene expression state space. And the question you want to ask is not just following the trajectories and you know what's happening, but also you want to know why this state go in the sort of the gene expression from this state to the other state, right? And that's the key. And you want to know why it goes, and not only it goes there, but why it goes there. And so this requires you to have some kind of a global picture to understand this, because you want to understand why it goes there. So it will be natural to imagine there is actually a background of a mountain like shape behind. So what that really means that if you can imagine this state corresponds to the bowl here, this state corresponds to the bowl here, instead of a flat land, you actually have a shape, mountain shape behind. Then what that really means that if you see that this state goes from here to here, you see why this state goes from here to here, because this is at the mountain top, this is the mountain bottom. That's why globally it has to go from here to here rather than going to somewhere else, right? So this is a way of understanding this. This tells you maybe a kind of global description is helpful, and it could give you some rationale of understanding of why the state goes from one state to the other in the sort of global understanding, a global way of understanding this. Now, this is all pictorial sort of a view or pictorial way of understanding this. The question is, can we actually put that into the reality? Can we establish something like that? Are we able to do something like that in the physical sense? Such kind of approach does exist in the equilibrium systems. That is to say that there are two ingredients in the equilibrium system. The number one is that the weight of the state is controlled by the interactions between the components. That's number one. If you know the interactions, you know the weights of the state. If you know the weights of each state, then you can see how you go from one state to the other, right? Because if you know the weight of each state from one to the other, you know this is the trend, because this is the low weight and high altitude, high potential, and this is a high weight, low potential. So that's why you go from here to here. You can see why this is so. Okay, this is called Boltzmann. The other part is that the dynamics, the next movement you're going to move is determined by the gradient of this weight. So this tells you what you are going to do next. So this is exactly the equilibrium state for the equilibrium state. And one could ask the question, are our systems in the study equilibrium systems? If so, then we can follow the recipe of the equilibrium thermodynamics of statistical mechanics and find out the whole description based on the interactions between, for example, genes. So we have a few examples using this idea, like the gradient equilibrium ideas, such as in the evolution, differentiation, development, protein dynamics, and protein folding. The last two, protein dynamics and protein folding and binding, are very successful. I would call this the evolution, the differentiation, development. These are the earlier approaches. These are very different. These systems are at the very higher scales compared to the proteins and protein folding and binding. Therefore, these systems are less likely to follow the gradient rule, although protein dynamics, protein folding, and binding are actually following this equilibrium dynamics rule, because they are mostly spontaneous process. So something like folding and binding can occur spontaneously. You don't need extra effort or outside support to do so. However, things like differentiation, development, evolution, and gene cell fate decision making, typically they require outside help. Okay. So my description about this is, if the potential of the interaction is known, then the force is the gradient of this potential. Everything is easy. Everything goes back to the equilibrium description. However, for most of the cases, most of the living system cases, this is not so. In the fact that if we go back to this equation, the x dot equal to fx, this represents a large class of descriptions of the evolution dynamics of the systems. The driving force for the whole system cannot be written as a gradient per se. This is the truth. And if you try different kinds of systems, you will see immediately, typically, this driving force is not a gradient. I mean, try your examples and see. And if this is not a gradient, then obviously it's another way of saying that it's not an equilibrium system. What can we do about it? Can we still have a global description about this system? So this is a little bit abstract, but I'm going to show you enough examples so you can see what happened. So still, what we have found over 10 years ago is that although the driving force cannot be written as a gradient, it's not a gradient like in general. It can be written as some kind of gradient force plus something else. And you would say, yes, I can always write down something as a gradient plus something else. However, we realize that this sort of additional force that we have found is not arbitrary. What it does, it satisfies certain properties. The first property is that it directly links with the non-equilibrium or non-equilibriumness. So if it's zero, for example, if this component is zero, this represents there's no net input or output to the system. Therefore, this goes back to the gradient. It becomes a closed system. If this is not zero, and this represents the net input or output or the flux of the system, therefore, it represents the degree of detail balance breaking, means it represents the degree of non-equilibriumness. Okay, so the another property that it has is this force, instead of gradient-like, it's actually rotational-like. This is like a rotational force. This is like a gradient force. Do you actually see any of this kind of combination in your education? This is the sort of the details in the mathematics. You don't have to worry about that, except that you can just remember that mathematics says that you have two. In general, the general force can be decomposed into two. One is the gradient-like force. The other is the rotational-like force. What does that mean? It means that, for example, if I want to study a genetic network, gene regulatory network with many genes, and this can be thought of as one particle moving in a multi-dimensional state space, right? And this is what we just show in the sort of pictorial view in that example. A equilibrium system, if the system is in equilibrium, then the analogy here is this gene regulatory network. Let me use that still as an example. It's like a charged particle moving in the electric field in the multi-dimensional state space. So the analogy here, the mathematics is also very much the same. However, if you have a non-equilibrium system, the dynamics of the network, for example, is like a charged particle moving in the electric field and magnetic field. So this is the key. And so you can imagine that you can imagine this, if I want to understand it in the global way, what a gene regulatory network does or evolve, it is actually analogous to a charged particle moving in the electric field and magnetic field. So that's sort of the picture. So also, let's forget about the mathematics and see what's the consequence of this. The consequence is the following. So if you want to go from one state to the other, this is very often happening in the cell fate decision-making, going from one state to the other. What's important here is the speed, how fast you go from one state to the other, and how you go from one state to the other path. So if you look at this, you will realize that if you just following the gradient, then you are going to pass through this saddle point, and going forward and going backward is the same path. And the speed is determined by this transition state, the free energy, the sort of the potential energy from this state and this state. The difference between the two determines the rate going from one to the other. However, due to the presence of this magnetic rotational force, then the path doesn't go from here to here or here to here. It goes, the rotational force sort of drives you going through some kind of a spiral way. So it deviates from the gradient path going this way. And going backward is different. Not only that, if you want to determine how fast it goes from one state to the other, then you have to use this as a reference state, as the new transition state, rather than the old transition state, right? Because you never even go through that. So you can write down the transition state theory based on the past. Now, the other thing is related to the so-called fluctuation dissipation theorem. So you kick one system and you watch how it relaxes. And in the equilibrium system, you kick the system, you watch it how it relaxes. It's equivalent of seeing the fluctuations near the equilibrium state. However, in the non-equilibrium state, if you kick a system, I mean, this is often happening in the experiment. If you kick the system, watch its relaxation, it's not just equal to the state, this steady state fluctuations. In fact, the difference between this and this will give you something, will be determined by the this net flux in or out, or the degree of detail balance break, or this non-equilibriumness. So statistical laws are changed. Also, one can see that the entropy production rate, this is something that we can see the total dissipation from the system and the environment, is not only used to fit for the free energy dissipation, but importantly, it will also be used to maintain the equilibrium, maintain the steady state. It's called housekeeping heat. So the entropy production is given for the dynamics, where you have the relaxation or dissipation from the free energy time-dependent process. And even in the steady state, you could have a chance to dissipate energy, because even you don't change in time, you can still have an internal flow, therefore dissipate energy. So certain part of the dissipation is coming from there. Okay, let me go and give you a real example and see why we are interested in this such a global description. Here, I just show you a cell cycle, a gene regulatory network, rather simplified one, for the cell cycle process. Why are we interested in this? Because if we want to go beyond the molecular description, then immediately we will ask, what's the next level? And furthermore, what is the fundamental unit of the living cell, of the living objects? Obviously, you will say, oh, cell might be the smallest unit for living creature. And we should understand the cell before we understand the organs or even creatures. So the cell as a sort of a unit of the living creature or living systems should be the one we should focus on. And this is exactly what we are going to do. And for the cell, there are several key ingredients. One of the key functions is that the cell can divide, right? And cell as the smallest unit of living systems has several important properties that signify that as the living objects. The cell division is the one. Why? Because cell division means one cell can divide into two parts, the two cells. And two cells can be further divided to four, and four can further be divided into 8, and so on and so forth, so that the generation can be passed. So this is the one definition essential definition for living system. What is a living system? At least the living system should have this property of generate its offsprings, right? I mean, that's the definition of living system. So this is the essential feature for the smallest unit of living system. We're interested in this because we want to understand what is the global view of this cell division, which is the essential feature of life, passing the offsprings. Okay, and from many studies that this process is tightly controlled by the underlying gene regulatory networks, and which is basically showing here several different cyclins become very important in the red represents the repression, and the arrow represents activation, and you can see at the different stages, different cycling CDK pair plays important role, and you go through different process through the stages of the cell cycle and go back. So we want to understand what can we do, how can we describe this process? Okay, so this is the process we want to describe. Well, first of all, on the left is the typical textbook illustration of how the cell cycle process has been going. And so it starts with the G0, G1 state, and through the checkpoint, you can get to the S stage or S phase, where you can go further and pass the G2 checkpoints and get to the M through the M checkpoint and M state, and eventually divide. The cell divides. And we want to understand in sort of a physical and global way how to describe this cell cycle, or to physically describe this cell cycle. And this is the one. After we have been looking at the gene regulatory networks that tightly control this process, we can ask, what's the global view of this process? And the vertical axis represents the potential potential landscape. Basically, it's the gradient aspect, right? And the two horizontal axes represents the typical gene expression, specific gene expression profiles, right? And specific genes, their expression levels. And if you look at this figure, this is the landscape, right? And we can do it mathematically and get this landscape. What does this landscape look like for the cell cycle process? The landscape looks like a Mexican head, irregular Mexican head. The Mexican head, we know that the center is high, outskirt is high, in the middle there is a ring. This is called a Mexican head, although this is an irregular Mexican head, but it is an irregular Mexican head. Okay, so what does that mean? It means that every state in the system experience some, because this is the landscape, experience some gradient force from this landscape. So it's the gradient force. So the gradient force, what it does, it attracts the system down to the ring, right? This valley, this ring valley, and eventually it will reach the bottom part, right? The lowest, the well is where it settles, right? Now, if this is the way it is, if globally that the whole system dynamics is controlled by the gradient, that's it, then you will be settled in the G0 and the G1 state. The cell will be settled in the G0, G1 state, and will never get out. It will never get out to the other states. And this is certainly in contradiction to the actual observation of the cell cycle, because cell cycle has to go through the cycle for division. So what comes out is that you absolutely need the other force in addition to the gradient. Gradient can tell you some stability, but you need another driving force, which is this rotational driving force, or we call it flux, to drive you going through. Once you are in the valley, because of the traction or the gradient nature, you tend to stay in the G0, G1 state. But in fact, it is this rotational driving force that pull you around and going through this checkpoint G1, G0, G1, S, G2, and M phase, and going back. So this is the division process. So this is the picture of, and also what I'm showing here is a picture, but actually it's all calculated from the model. And this is something that we are not drawing just per se. This is actually based on the calculations. So this is the sort of the picture. So globally, you get attracted to the oscillation range, and once you are on the range, the dynamics or the dynamical motion along the range is dictated by the rotational force. And so this rotational force is necessary to generate a coherent oscillation or division for the process, essential for that. Okay. So that's very important to point it out. So one of the things that we realized everything corresponds to the biological textbook description, because the G0, G1 state represents a basin of attraction here. And S, G2, and M phase all represents local basins of attractions. And this checkpoints corresponds to the sort of the local transition state or the ridge connecting between the basins of attractions. And so, and the flux drives this through to go through the cycles. So this is the sort of the process. On the other hand, another thing which is very worthwhile of saying is that you may wonder, what is this mysterious rotational force? Where does it come from? And actually, we can show that. I'm not going to show you in the too much mathematical details. What we can show is that the origin of this rotational force is from the nutrition supply. So for the cell, if I don't give the nutrition, no enough energy, the cell will stay in the G0, G1 state, will not continue for the division. Only when you have enough nutrition or energy supply, you can get excited and going around. So this rotational force is linked to the nutrition supply. So I can also say that if you don't have this enough nutrition supply, then you cannot go around. So you cannot have a cell division. And if you don't have a cell division, you don't pass the generations, you lost the essential feature of the living system. Therefore, you are not a living system anymore. So from the single cell level, there's a threshold for life. If you have enough driving, if you don't have enough driving force, you're sitting down there. There's no life, no living system. But if you have enough energy supply, you can jump out and start going through this process called replication, or passing your life to the next generation. So this is saying this is not only important for cell division process, but also essential for single cell life. So that's basically what I want to say. Because let me, and the picture is always good. So if you have a low nutrition supply, as I said, you tend to be sitting in the G0 and G1 state. And if you keep on increasing the nutrition supply, eventually you are able to go around. But if you don't have a nutrition supply, you just sit there, and you're sitting there comfortably in a stable state. Let's consider a few other examples where cell can do a few things. One, I don't know. Can you see that now? Yeah.
Yes.
Yeah, okay, cool. So one of the things that we talk about is the essential thing for living is the division of passing the replication, passing the genetic information to the next generation. The other one is very important. The cell can do is cell can differentiate, doesn't change the genetic context, but it does change to a different state, which is very important, right? So this is this so-called differentiation. So cell can go from the stem cell to the differentiated state. Okay, so a while ago, Waddington in 1950s asked his friend, artistic friend, to draw a picture of understanding this differentiation and development process. So he envisioned that there's a marble at the beginning, at the stem cell state, that this marble gradually falls down to different trenches or different valleys, and that represents the different differentiated states. So this is so famous, so-called Waddington landscape. Although Waddington has this vision, and in this picture of what happened at the differentiation process, there's no quantitative or physical basis for that. For example, people don't know what that means, and what does the landscape means, what this axis means, what that axis means. Is this picture right or wrong? This is a metaphor, and the people don't have this kind of basis. Now we can base on the information gene regulatory motif or networks, we can quantify the underlying landscape of this process, of differentiation process. For example, the typical motif of the gene regulation motif for the differentiation process can be sort of the core motif can be described as mutual repression and self-activation. And based on that, we can quantify the landscape. And this is essentially the composite gene expression state space. And the other axis represents the self-activation strengths, which keeps on changing during the developmental process. So this is you can think about this is the axis representing the regulation change during the differentiation development represents this axis. And this axis is the gene expression axis, this gene expression axis. So after we look at this problem, we can quantify this landscape as well. So these are all drawing based on our approach, on the theory. But if the model can be used to describe this process, so this is all based on the calculations. So what happened is that now I do know exactly what this means. This is no longer a metaphor. You can calculate, and then you can calculate this For example, this at a large self-activation, you have a middle state which is stable, and as the differentiation process progresses, this landscape shape keeps on changing until the very late stages where you have a the state branches out or differentiated to the different basins of attraction. This corresponds to the differentiated state. So this Waddington landscape is not a static landscape. It's an evolving landscape. It's a landscape at certain stages. But as the differentiation of development progresses along the way, then the landscape shape changes. So this stem cell state gradually goes from the local stable to become globally unstable, from low to high, and eventually branch out to the new local basin of attraction, which corresponds to differentiated states. Okay. Let me mention that we can apply this to cancer, a differentiated, a more complicated differentiated state. We can also apply this to cancer. I don't have much time to talk to you more in details, but except that we can get a landscape for cancer where you have apoptosis state, normal state, and cancer state. And so essentially what you are interested in looking at is that you want to be able to, if you start from the normal state, you want to avoid to get into the cancer state. And if you are in the cancer state, you want to be able to get into the, easily get into the apoptosis state. That could be measured. The degree of difficulty can be measured by the sort of this barrier height in between the basins of attraction. So this is the normal, this is the cancer, this is the apoptosis state. Yeah. And let me not talking about too much about aging, which we have an application for that, except that I will briefly mention about a possible application to evolution. And the evolution, as we understand that in the Darwinianism language, is called a natural selection, or selection and adaptation. And so fittest survive. And this has been quantified by Wright and Fisher in 1930s and 40s and 50s. And basically, we can think about this is essentially a landscape picture, except it's upside down. So we in physics, we usually look at a potential landscape where the weight is inversely related to the potential depths. Here, the fitness is like a weight. So you are always going to high rather than low. I mean, but it's equivalent of the high fitness is basically corresponding to the low potential. So and this kind of a Darwinism fittest survive is, as I said, it has been already formulated by Wright and Fisher in terms of the fitness or adaptive landscape. However, there's a guy came out and criticized their approach. Basically, he's saying, well, this approach is all nice, except that what if I consider this case, essentially he consider a co-evolution process where you have a wolf and a sheep, and wolf obviously eats sheep, but if the sheep are all gone, this is not good because the wolf will be starved to death. So in fact, if the sheep number is down, the wolf number will be also down. Of course, if the wolf number will be down and the sheep number will be up, because you don't have the wolf around to eat the sheep. And eventually you go through this cycle. This is called co-evolution. He said if the co-evolution, in the co-evolution case, it doesn't seem that the evolution ends when you get into the optimal. In fact, he questioned even if you have optimal, because this is like Escher's case, you keep on running, like the co-evolution, you keep on running cycles, and how do you reach the absolute optimal? This is actually criticized by Van Valen, a Chicago evolution biologist, of this feature. So we realize that this is, in general, the evolution dynamics is not determined by the fitness landscape. It's actually, there's a missing component in the general evolution dynamics, which is to say evolution is not only following the fitness landscape, it also has this rotational driving force. So in fact, if you think about the evolution, it cannot be just going directly to the goal. It has to go through a spiral way. So that's the sort of conclusion. Let me not give in too much of the mathematics, but by just saying that if you consider there's an additional driving force, like a rotational driving force, you can explain this kind of Escher staircase, which is essentially the co-evolution case, where things go through the cycles. We have applied this approach to many different other branches of biology and also physical system, which I listed here. If you are interested, you can go ahead and look at our two review articles on this, one published in Advanced Physics, the other published in the Review of Modern Physics. Now, let me, in the remaining time, a few minutes, let me just mention that what we are talking about is not completely just theoretical wonderland. In fact, all of this we talk about can be measured in the experiment. So I just very briefly give you a few examples where both the landscape and this rotational force can be measured in the experiment. So the number one example I want to show you is the example of classic molecular biology example, where it's called a lambda phage, where these two genes mutually repressing each other. And we can, this is a very typical cell-fate decision making. You can go either one way or the other. So you think maybe this is the simplest one. So some people call it hydrogen atom in the molecular biology. What can we do with it? And we use it in collaboration with the experimental group in Johns Hopkins University. We actually did something technical, which is basically, in order to see these two genes, we label them with the fluorescence proteins. But if we label with the two different fluorescent proteins, maybe they have different maturation times. You can't see them simultaneously. So a technique can be developed to use the same color, but different location in the cell. One is in the middle, one is attached to the membrane. So you can see two different kind of gene expression patterns. One in the middle, one is on the side of the cell. So the interesting thing is, what do we see? Different colors representing different states. This is this one gene C1, one is the crow, and when both are high, you have this purple. When both are low, it's the red, and when one is up, it gives you the blue, and the other one up, the other one, the current one is low, is representing the green. So you can see, actually, this is the time. The state actually goes from one to the other, switch from one to the other. pretty often, actually, in the time observational window. They keep on changing the color. And if you do the statistics of these real-time observations, you can see this graph. Instead of 2 states that we expect, you actually see 4 states. And you see this state, this state, this state, and this state, clear 4 state. So in other words, this mutually repressive gene circuit, like lambda phage toggle switch, one naively say will be 2 states, right? 1, 0, and 0, 1. But in fact, you have 4 states here. And not only that, let's see. We also see that unexpected from what we think before, that one goes from one state to the other directly, it actually goes through these bridges much easier. If we don't see this real system, it doesn't, we always think it's just 2 states and they just go directly from one to the other switching. But actually the switching we found is actually through this bridge. They don't directly go through one another. So this tells you a lot of things, but this gives you the landscape, right? This is just the statistics of fluorescence give you exactly the quantification of the landscape. So this is the 4 basin subtraction landscape representing 4 states. One last example is that for single molecule enzymatic dynamics, we all know that there's a Michaelis-Menten law. Basically, it says that the rate of the activity is inversely proportional to the substrate concentrations. And we consider a single molecule enzymatic reaction dynamics which catalyze 2 sort of systems, right? And through the horseradish peroxide, it catalyzed two systems. And I'm not going to go through the details, but just tell you the sort of essentially the conclusion. The conclusion is that that if you have the equilibrium system, then we recover the Michaelis-Menten, the normal Michaelis-Menten rate. But if we are in the non-equilibrium state, then you are going to deviate from the Michaelis-Menten rate. And this is all, if you break the detail bounds, then you have this rotational flux, which is representing the rotational force part. So you can observe the deviation from the Michaelis-Menten, and this will immediately tell you how big this rotational force is. And in the experiment, we can see that the straight line is the normal Michaelis-Menten rate, and this is the measured one, the rate, which is certainly, it's not a straight line. This is the Michaelis-Menten prediction, and this is the real data. It's very much deviated from that. So from this, we can actually find out how big this rotational force is in the experiment. So let me go, I know I'm going beyond. So let me summarize what we have found, that we can compare between the equilibrium and non-equilibrium approaches. The essential part, you don't have to look at through all, but the essential part is that we found that the driving force for the global dynamics, including the gene regulatory networks and others, or the system, all different levels of the system are controlled by 2 forces. One is the gradient of the landscape, and the other is the rotational driving force, which is the sort of the flux force. And they have many different consequences of the, as I mentioned in the cell cycle, it determines the threshold for single cell life. And for differentiation, determines the differentiation path and the reprogramming path. And for the evolution, it determines the co-evolution. So let me thank my collaborators for the landscape series, mostly from Stony Brook and the two and the experimental group in collaboration with the Chinese Academy of Sciences. I want to thank you for your attention. Thank you.
Great, Jin Yang. Thank you very, very much. You know that I have been following your research, and it clarify a lot of questions that I have in my mind. I have maybe one or more questions. One of them is the following: if we have the landscape, we can determine the trajectories. Okay? Can I do the inverse? If I have the trajectories, can I determine the landscape like a reverse engineering method?
Yes, I think I don't have the time to show the details, but for example, in the experiment, I show you the toggle switch, this lambda phage. This is how we determine the landscape based on the real-time trajectories of the two genes. One is the C1, one is the crow. The way to do it is to do a joint histogram or joint statistics of these trajectories. Essentially, the C1 and the crow, you have real-time trajectories of these two genes, and I do the collect statistics or the histograms of these two species or two genes in the steady state, and then that's how I show this graph of 4 basins of attraction there. That one is actually just the pure statistics collected from the two gene trajectories. It's just the joint histograms of the two genes at the steady state directly from the trajectories. Yeah.
Great. I will make just one question more. I understood that the landscape in the non-equilibrium and in the equilibrium situation are the same. Just the behavior of the system is different. Is that correct or not?
I think it's not quite, because what happened is that in the equilibrium state, you can go from the interactions and directly through the Boltzmann relationship and get the probability distribution, equilibrium probability distribution. In the non-equilibrium systems, it's the other way around. The logic is the other way around. You get the steady state distribution, like what we just mentioned or discussed. Essentially, if I go to the experiment, I observe some trajectories, I get the collective statistics, and the joint statistics give you, histogram gives you the landscape. Then we can just get the landscape. But this is not necessarily the equilibrium landscape, because it just means you are at a steady state. The steady state doesn't guarantee you to be in equilibrium, because you have this possibility of local internal flow or internal rotational flow around, so that even though the weight doesn't change of each state, you could imagine that, like a baseball, each guy is sitting in the base, but they pass through the balls around. And if the passing A to B and B to A sort of in the same pace, then it's in equilibrium. But however, you could have a situation where A passed the ball to B, B passed the ball to C, and C passed the ball back to A. And this flow is forever flowing around. And although the state A, B, C doesn't change in time, but there's the internal flow always passing this little ball around. So this is what we call that steady state. Steady state means that probability doesn't change, but there could be an internal flow going A through ABC. So passing the ball from A to B, B to C, C to A. So they are actually different, the non-equilibrium system and the equilibrium systems.
Great. Marcos Vieira wanna make a question?
Yeah, go ahead.
Go ahead.
Am I vendo?
Yes.
Yeah.
Hi, Wang. I would like to thank you for your presentation, and I'm currently working with these kinds of modeling, and I think that if you could talk about the relation of increase in entropy in cancer states with the dynamics inside the cancer attractor and the possibility of observing ergodicity. And another point is a possible analogy with cancer and the evolution and selective pressure. I don't know if I was clear.
You want me to comment on the connection to cancer and evolution? Is that what you want me to do? Or?
Yes, first about the increasing entropy observed in cancer states, and its relation with the dynamics inside the cancer attractor, and if this can lead to a possibility of observing the ergodicity in the system.
I think I think maybe you're referring to the I would say it kinds of natural for me to understand this, because the cancer is, well, by itself it consumes a lot of energy because the nature of cancer is to grow, so it consumes lots of energies. So in the entropy language, it generates a lot of heat, or the entropy per se. So the entropy generation or the entropy production will be immense. So I would expect that entropy will be produced or generated, or whatever ways we can say. So yeah, I would expect compared to the normal state, cancer state consumes a lot of more energy, and the entropy production rate is expected to be higher. I mean, that's my expectation, although, I mean, this probably, we have to calculate that through. So the quantity that we typically use to measure this is the entropy production rate, the total entropy production rate for the system. That's sort of the things that a non-equilibrium measure of these, you know, a marker. I would think it would be a marker. For the cancer evolution, yes, because you know, the growth and the fitness, and this is all sort of the related aspect. How do you sort of measure the fitness? And so in this language, I would say there are two things. One is the cancer itself has landscape because the gene regulatory networks. The other is they have to sort of fit the environment. In other words, in the cancer regulatory network, the links may not be as robust. They are actually the links, or this regulatory strains have been will not be kept as constant, might be able to change. So in that sense, they can adjust the strengths of the interactions among the genes or by mutations to effectively change the regulations among the nodes to have the better fitness. So in that way, you could have a sort of a cancer evolution to have a better fitness or growth.
Thanks. And due to that fact, we observing a single cell RNA sequence that the expression level is almost full in some region of space, and I'm thinking if the freedom of the cancer states will not lead to some ergodic trajectories, so that it's a lot free to goes to the phase space, you know. I don't know.
Yeah. I think there are a couple of issues here. I mean, these are all belong to the cell fate decision-making process. I would say for single-cell RNA-seq data, the ones that one can do better is the differentiation and the development, because the process is relatively short. In other words, for example, differentiation, if you choose the right creature, is it could be just 7 days or 2 weeks. So you can do the single cell RNA-seq in this, say, for example, 2 weeks, you can cut 10 time points, and you can get the data relatively straightforwardly and not so much time. However, if you want to study the cancer, for example, the process from normal to cancer state, this kind of cell fate decision-making process, typically the time is long. I mean, you're not going to just wait for 2 weeks to get the cancer, although you may be able to accelerate the process. For example, the mouse, and like 2 weeks it grows up to the cancer. It depends, but for the normal situation, it takes too long. But maybe for some animals, you can accelerate this process. You can still do single cell RNA-seq. I'm not sure if people have done that, so that in 2 weeks you've grown up to cancer. So in these 2 weeks, you can cut the time points and you have the single cell RNA-seq. I would actually expect if you collect enough data of single RNA-seq, you may be able to get some feelings about this cell fate decision-making process. I mean, people are trying to, now this is an active field, trying to map out the underlying landscape and from the data, and try to figure out how you go from one state to the other. I think for differentiation and development, the data is richer and more well documented. It's probably easier to deal with than the cancer. But maybe the cancer field is catching up, but they have to do some accelerated ones rather than the normal one. It takes too long to develop the cancer.
All right. Thank you very much.
Yeah.
Great. Thank you very, very much, Jin Yang.
Oh,
it was
thank you.
a pleasure to have you
My
here.
pleasure.
Great. And we will stop now for lunch, and we will be back at 1:30 Brazilian time. See you there.
Bye-bye.
Hello, Thomas. Hi.
Can you hear me?
Yes, yes.
Excellent.
Just coming out from the class. Hello, Thomas.
Hello.
Hi. 1 second here. And echoes. Espera un segundo, there is some echo. Just coming out from the there is a YouTube somewhere. 1 second.
I think perhaps it's me.
There is an infinite cascade.
There you go.
1 second, if I can manage to find the YouTube. I don't know where it is. 1 second. Killing on YouTube, I think it's okay. No, it should not be. Okay, so I am. So we should talk some other time. Did you find that link?
Yes, actually, I wanted to write you about that. This dirty thing is an industry thing, so it's not exactly a science thing.
Because we did something similar with Belgium. It was scientific. So I will look at it.
I need to. I haven't looked at in detail at the call, but this they usually is I mean, these guys usually want you to involve companies and have industrial partners and things like that.
Because it's strange. Because from our side it's not, but
I need to look at it. But you know, I'm not very hopeful. But I will.
Because for the Belgium, it looks like that, and then I wrote. But could be epidemiology model data, and she was yes, of course, but it was not clear from the call. So it's better to ask because sometimes it's praised.
I will have a look, and I'll also ask.
Yeah, someone from there, so I can ask again because it was not clear for me as well. So we can try to. You like to test the?
Yes, I can try to share. Let's see.
I see very much.
There it is. Share. No.
Great. Okay. Very nice picture.
I don't see anything in my screen now. Okay.
No?
No. Okay, let me see.
It happens sometimes if you don't have Zoom installed, or you just
No, I have it, but yeah. Okay, anyway, you can see the.
Yeah, we can see there is how to say,
yes,
it's not completely full screen. There is also you can see the slides, but it's okay. I mean,
now it should be full screen now.
Yeah, but you see
Let me try something different.
It happens to me in the PDF. You have to close that column.
Because I think that if I put it first in presentation mode and then I share, I think that then you should see. What about now?
Yes, now it's perfect.
Now it's okay, good.
Yeah, now it's very nice picture, by the way. I'm jealous.
It's I found it in Google, so it's
yeah, very nice.
it's a polymerase replicating DNA.
Yeah, I can see that.
I like it. Yes.
Yeah, very nice.
Okay, so I don't have links or anything, so it should be okay.
So we wait 5 minutes still.
Yeah.
People are resuming lunch. So how things are being there?
Well, now it's extremely hot.
Yes, I heard. I supposedly have to travel to Europe.
Bring the air conditioning with you.
Not immediately. At the end of the first week or beginning of the second week of August. Yeah.
Of when?
August.
So are you going to Italy or?
Yeah, to see my mom. This is the idea. I know August is supposed hotter even. I can imagine.
Here we are now in this canicula period. From the 15th of July to the 15th of August. In Spain is the hottest time of the year.
Yeah.
At the end of August, it's supposed to improve a little bit, but I'm not sure if that's going to happen.
Otherwise, COVID-wise, because here you see here we got the waves after you, so it's like still.
Here is we are now in the 7th wave.
Mm-hmm.
But so there is quite a lot of contagions and virus going around, but it's not really hitting people hard. So it's kind of like a flu, more or less.
No, because here is not well. Here maybe. Because of the vaccine and everything, it's true. We have much less how to say death. No, there are more people get infected and less death. But we have some cases this year of people being affected intellectually, neurologically. Maybe there are not a lot, but we can see that some people affected their intellectual capacity.
No, here I haven't heard about this. What it's now, there are a lot of news about this persistent COVID thing. You know, these people that keep on feeling bad for months and months and months.
Yes.
And we have had quite a few cases of that, and so that's. But I haven't heard about people.
No, I have even around at the university, you would see some people that you know before and after. Of course, as I say, they are not a huge number, but there are some cases that are kind of extreme. So the person doesn't have the same way fast of thinking, and got exhausted if he studies more, and so we some person even come to me and told me, among the students, and I had the same student before and after, and so it's something that we observe, but this I don't know, how is the percentage of this with respect to COVID, but this happened. We see some cases, yeah. And then I would like to know if it's a permanent effect or not, or is temporary. This was my question mark. This was the question mark. Yeah. So I guess that 1 second. Let me check, because the organizer, they were having lunch. It's a type. So let me see the other one where they are. See, 1 second. Checking. And down, yes. Okay, so yes, just a few minutes, then we can start. So Isabel is no longer with your group?
No. Isabel was let go.
Because, we advanced with that project. We have very nice results here.
Oh, great.
Yeah.
She's now, that she had a part-time position at the supercomputing center.
Yes.
And she was offered a sort of a full-time job there. So
Right.
I don't know. I haven't talked to her for a few weeks now, so I don't know. She told me that she was going to sort of think about it until September, and that in September she would take a decision of whether to take this full-time position or stay.
Yeah, but we'll be. I mean, I think we have some nice results, about what we so that.
Well, I mean, you can still contact her.
Yeah.
So that's not.
But it will be interesting, since started CRM, to try to think about some common. But yeah, so some other time we can talk about that.
Yes.
Okay, so I guess we can start. So,
whenever you say.
Yes, 1 second. Okay, so welcome to everyone. The fact of being blind, and now people are more blind than in person, we have to use again to socialize in person. But anyway, so welcome to everyone. It's a great pleasure to have Thomas Alarcón, with us again from Barcelona. Are you in Barcelona, Thomas, in this group?
Yeah.
This time currently in Barcelona. And actually, the first time I met Thomas was in Paris. It was, I think, 2004, 2005. So it's a long time ago. And since then, of course, it's been many success. It's been appointed ICREA research professor. He's a ICREA researcher, and he's at Serum, is where he's the head of a group of, 1 second, I forgot, the biological mathematical computational and mathematical biology group, a very active group at CRM, and also is a member of the scientific committee of the Catalan Mathematical Society. And it's part, of course, of the Barcelona Graduate School. So it is a great impact in interdisciplinary research. So Thomas, it's a great pleasure to have you with us again. Thank you.
Okay. Thank you, Stefanella, and all the organizers for the invitation. And So I'm going to talk about today some recent work that I've been doing in collaboration with Josep Sardanyés, which is a colleague of mine at Serum in Barcelona, Toni Guillamón, who is a specialist in dynamical systems at the Politècnica de Catalunya, and he's also now affiliated to CRM, and also with my experimental colleague and friend Javier Menéndez, who is at the Catalan Institute of Oncology. Okay, so what I'm going to talk about today has a lot to do with gene expression. Okay, so gene expression is a set of mechanisms that take care that genes are expressed, that the genes are transcribed in an orderly and timely fashion. Okay, and the mechanisms for gene regulation are diverse. There are several modalities of such a process. One of them is perhaps the best known is regulation by transcription factors. Okay. By the way, can you see my pointer?
Yes, we can.
Excellent. So, so yeah, regulation by transcription factors. So transcription factors are special types of proteins that have the ability to bind special regions on the DNA molecule, which are called promoters. Okay, so these promoters, when they are bound to the proper transcription factor, they are allowed. So they allow the DNA replication machinery of the cell to bind to the initiation site of the gene, and it triggers the expression of the corresponding. Sorry, say again.
No, it was just.
Okay. So yeah, I mean, in this particular case that I'm showing here, basically this is a self regulatory gene where the protein expressed by the gene is basically the main component of the transcription factor. Okay, so this would be a self-regulatory positive feedback loop of gene expression. Okay, but you know, besides this regulation by transcription factors, there are other types of regulation, and in particular, the one that I'm going to be talking a lot about today is epigenetic regulation. Okay, and epigenetic regulation has to do with whether these transcription factors are actually allowed to physically interact or reach the promoter regions. Okay, so as you know, the DNA is a very long molecule, which, in order to fit in the tight and close space of the nucleus, needs to be coiled and around and super coiled and organized in such a way that it can fit within that space. Okay, and that poses a problem for transcription because, for example, in this example here, or perhaps perhaps here. Okay, so here this region that we can see in pinkish represents one of these promoters. Okay, so if the DNA is packed in this configuration, this promoter region is sort of hidden from the transcription factor. So the transcription factor cannot access this region because the rest of the DNA is sort of in the middle and it cannot reach it. Okay, so if the DNA is in this configuration, even if there are transcription factors around, the cell cannot be transcribed. Okay, so we will refer to this situation as silenced, as a sort of that the gene is silenced, it's epigenetically silenced. Okay, whereas if the DNA is in this configuration, okay, so now here this promoter region is now in the open and is accessible to the transcription factor. Okay, so in this configuration, if there are transcription factors around, the gene can be transcribed because they will be able to reach physically the promoter region. Okay, so now the issue now is how do we go from here to here? Okay, and this is basically what epigenetic regulation is doing. And in a nutshell, and very briefly, without inundating you with biological details, this happens because so well, let's go one step back. Okay, so we were talking about this coiled structure of and packed structure of DNA. So this has a different number of levels, and the level that we are going to be interested here is the level of chromatin. Okay, so chromatin is the concatenation. So it's this structure here. Okay, and it's the concatenation of these units called nucleosomes. Okay, the nucleosome is a unit formed by 8 proteins called histones. Okay, so here you can see 4, but in the back of these ones there are another 4. Okay, so there are these proteins are histones, and the nucleosome is completed by the DNA wrapped around this octamer of the histones. Okay, so these histones have tails. Okay, and these tails have different residues. Okay, so what the epigenetic regulation does is basically adds modifications to these tails. Okay, and depending on which modification is added to which residue within this tail, the chromatin can go from being in a packed state like this one, where promoters are not accessible, to loss to a configuration in which the DNA packing has loosened up a bit, and the promoter is accessible, like in this situation. Okay, so for example, this would be this could represent one of these molecules that's a polymerase or something that starts the gene expression process. Okay, so basically, the only thing that you need to remember is this: that there are some modifications that pack the DNA tighter, and they don't allow the promoters to be accessible, and therefore they hinder gene expression. And that there are other modifications that open up that structure and allow the transcription factors to access them. Okay, and the other thing that it's important for the model that we will see later is that these modifications are added or removed by specific enzymes. Okay, so this is an enzymatic reaction in which an enzyme sort of collocates the mark in the proper place. Okay, so this is epigenetic regulation sort of in general, in a nutshell. I hope it was more or less clear. Okay, and there are different types of epigenetic regulation, and the one that I'm going to focus on today is a specific type of epigenetic regulation that it's called bivalent chromatin. Okay, so bivalent chromatin is a specific type of epigenetic modification. Okay, so it's specific to one of the histones in this nucleosome structure that I was talking about. So this the third of these histones have a tail, and this tail contains 2 residues called lysine 4 and lysine 27. So we will refer to this as K4 and K27. Okay, and if the K27 residue is modified by a 3-methyl molecule, and this is a negative mark. Okay, so if the promoter is dominated by this mark, this means that the chromatin is packed tightly around the promoter, and the transcription factor cannot reach the promoter region. So there is no transcription. Okay. If the one, by contrast, if the one that is trimethylated is the K4 residue, then this opens up the chromatin and allows the transcription factor to reach the promoter. Okay, so if the promoter is dominated by this mark, then the gene can be transcribed. Okay, and then there is a third state that it's the biology referred to it as poised, in which both marks are present. Okay, and in this configuration, the level of gene expression is still low, but it is called poised because in the presence of the enzymes that remove the negative mark, this immediately goes to an open state and the gene can be transcribed. Okay, so this is something that it's very common during development. Okay, so this is a way in which specific genes having to do with differentiation in different types are kept silent until they are needed to produce the specific type of cell that they need to produce. Okay, so that's the thing. So basically, what we are going to look at is this circuit of gene regulation, a self-regulatory gene, where the transcription factor is essentially the protein that the gene is producing. This is very common in embryonic development. This kind of positive loop, because this allows rapid changes of state. Okay, and what we are going to look at is the influence that this system of epigenetic regulation has on the transcription of the gene. Okay, if the promoter is mostly modified in this way, then the gene will be closed, and the other way around. Okay, if the dominant mark is this one, we are going to allow the gene to express itself. So basically, what we are going to formulate is a stochastic model in the form of a continuous time Markov process, where we are going to track the amount of negative modification in the promoter, the amount of positive modification in the promoter, and the amount of transcription factor in the system. Okay, so that's what we are going to do. Okay, so this system of Divalent Chromatin was first investigated in the context of embryonic development, as I was saying, but recently has emerged as a epigenetic trait in cancer, and specifically as a druggable therapeutic target. Okay, so for example, as we say here, so different regulators have been identified as targets for cancer therapeutics. So for example, this enzyme here that is responsible for adding the negative mark, okay, and these demethylase, which are responsible for removing both marks, have been identified as possible, have been found to be dysregulated in a number of cancers, and therefore they have been proposed as targets for new drugs. Okay, and also very importantly, and this connects somehow with Jin Yang's talk just before lunch, these epigenetic these regulators, the Divalent Chromatin regulators, are key factors in something that is called epigenetic plasticity. So this means that the barriers between the different states, the different differentiation states, can be lowered, and this often in the context of cancers leads to pathological plasticity. This is the fact that cells can change their identity just by sort of random fluctuation, and also to cell reprogramming. And this is what it's interesting from the point of view of cancer, because cell reprogramming basically means that cells become more undifferentiated, more stem cell-like, and they can acquire stem cell properties, particularly self-renewal. Okay, and these regulators have been sort of identified as mechanisms for these things. Okay, so I'm not going to go into the details of the model because it's quite complex. It's been published, so if you're interested, you can look at the paper, or you can just drop me an email if you're curious about it. Okay, so but let me just describe it in a few words. So as I was saying, this is the system that we are going to model. So basically, we have a self-reinforcing transcription factor, self-reinforcing because the protein product is the transcription factor. So the more protein product, the more transcription is going to be. Okay. So then we are going to also to model the abundance of the positive mark in the promoter of the gene, and the amount of negative mark in the promoter of the gene. Okay. So if there is more of this than of this, then the transcription factor is allowed to act and trigger gene expression. And if the dominant one is this one, then the transcription, the access of the transcription factor to the promoter is blocked. Okay, so then also the collocation of these marks is also reinforcing. It's also, there's a positive feedback. The more there are, the more the larger the rate of collocation of these marks, and this goes both for the positive and for the negative mark. Okay, and quite crucially for our model, it's been also identified experimentally that transcription factors inhibit the collocation of negative marks and help to the addition of positive marks. Okay, so okay, so as I said, the model is quite complex, and the analysis is also, well, it's a bit involved, but we can, I mean, even if the model is quite complex, we can take advantage of separation of time scales. Okay, so basically what we have after doing some analysis and looking at experimental data regarding all these elements, what we have, so the model basically assumes that there are sort of fast variables, which are the epigenetic enzymes and the complexes transcription factor promoter, and this evolved very, very fast. And by doing a quasi-steady state, stochastic version of the quasi-steady state approximation, we can find analytical expressions for the probability density functions of these quantities. Okay, so this leads to a system that it's still quite complicated, but so there is, once we have eliminated these variables, we still have a system with a slow fast dynamics, and we can do another round, say, of quasi-steady state approximation of elimination of the fast variables by means of singular perturbation analysis. Okay, and after all this, what we have is a system that we can solve analytically for the average master equation of the transcription factor. Okay, and in fact, what we have is an analytical expression for the density, for the probability density function of the concentration or the abundance or of the transcription factor. Okay, which is given by this this formula here. It's, again, I mean, this is a deceptively simple description because there is quite a lot of work to do in doing this sort of hierarchical elimination of the fast variables. Okay, but again, I mean, this probability density function is characterized by this function u, which we call a quasi-potential, okay, which is given by this function here, and all these quantities are quantities that we can calculate analytically and come from this multiple scale analysis that we have been done. Okay, so and this is quite nice because now everything that we need to know about the long time behavior of the system is contained in this function here. Okay, and this function here is a function of the concentration of the transcription factor, depends on this parameter here, which is the system size, which is, it works like the inverse of the levels of noise. The bigger the system, the less noise we have in the system, and quite crucially also depends on a bunch of parameters that have to do with the activity of the enzymes. So how good or how bad are they at adding or removing the epigenetic marks that open or close the transcription factor. Okay, so so again, I mean, this dependence here on the parameters. I have made it explicitly because this is going to be quite crucial to deal with intratumor heterogeneity. So we have heard earlier today a very interesting talk on drug resistance. Okay, so the take that I'm going to take on drug resistance is slightly different to the one that Rafael, I think it was, told us about earlier, right? So here I'm going to focus on heterogeneity. So intratumor heterogeneity, I mean, this means that cancer cells are not, I mean, even if they are clonal populations, so they share the same DNA, they behave differently because of differences in the metabolism, etc., etc. So there is even within a tumor there are many different cells or cells behavior. Okay, so and this is a huge barrier to drug to treatment of tumors with targeted therapies. Okay, because these targeted therapies usually are directed to a specific molecular targets that have been discovered to be important for cancer cells. Okay, but so in the face of it, it looks like a good idea to try to find drugs that target these molecules. But unfortunately, as it says here in the abstract of this paper that I am showing in this slide, most cancers eventually acquire resistance to targeted therapies and end up relapsing. Okay, and why is that? Well, you know, it's the sources of heterogeneity are quite complex. So I'm not going to go into that. But the thing is that if you are targeting a particular molecular element in cancers and in cancer cells, and the population of cancer cells is heterogeneous with respect to that particular target, you're going to have what they call here evolvability of de novo resistance. But what this means is that in this heterogeneous population, you're going to have cells that are going to be very responsive to the drug, and those are going to be cleared immediately. But you're going to have cells that respond very poorly to that particular drug. Those are going to linger around, and eventually they are going to repopulate the tumor. Okay, so we need, so when one is trying to predict whether a specific molecular target is going to be effective regarding a specific treatment, one should always worry about how a heterogeneous population is going to respond to that drug. Okay, and to do that, we need to somehow model heterogeneity. Okay, and what we have sort of developed in the last few years is what we call the parameter ensemble approach. Okay, so basically this consists on a series of steps, which I'm going to describe now one by one. So first, we need to establish what are the possible behaviors of the system in terms of the mathematical analysis that we have done. Okay, so I will be more explicit about what I mean by this. Okay, in a minute. So the second thing that we do is, once we have established the different behaviors that we can have in our model, we need to explore the parameter space and associate to each one of those behaviors that we have identified. We need to see in the whole space of parameters which parameter sets are the ones that are compatible with a particular behavior. Okay, so we create in this way we create these subensembles that are again compatible with this. Okay, and they had, so the aim or the hypothesis behind this is that the heterogeneity within these subensembles somehow captures the heterogeneity within the cell population. We will see an example in a minute. Okay, now, of course, the first step to do this is that we need to use now experimental data to match these behaviors to biological features or to cellular epistates or to whatever we have data about. Okay, so in this particular case, what we want to identify is which one of these behaviors and which one of these subensembles associated to the behaviors are associated with cancer-like behavior and with normal-like behavior. Okay, because that will start to giving us clues as to which parameters are important to try to transform the cancer systems into the normal systems. Okay, and again, I mean, well, this is basically what I just said. I mean, then we need to analyze these ensembles and find search for regularities, biases, correlations, or whatever that could be exploited to formulate either combination or single target therapies. And last, use the example heterogeneity to somehow assess the effects of the proposed drug combinations against the background of the heterogeneity of our subsystem. Okay, so I'm not going to go now through each one of these steps. I'm going to skip a couple of them in the interest of time, of course. We're going to be here until tomorrow. So the possible behaviors. Now, this is almost a trivial exercise because, as I said, everything that we need to know about the long-term behavior of the system is contained in this function here. Okay, and what we have seen is that basically this function can be, this quasi-potential can be either monostable or bistable, and that's it. Okay, so the monostable situation can be divided into two cases, where the monostable, where the stable, where the minimum corresponds to a low concentration of the transcription factor, or to a situation where the minimum of the quasi-potential corresponds to a high expression level. Okay, and then there is the bistable, where we have two states which typically correspond to a possible state with low gene expression and another one with high gene expression. Okay, And then we have this third situation, but this fourth situation, but I'm not going to talk about this today. So as far as we are concerned today, so we have these three behaviors: low gene expression, high gene expression, and bistability. Okay, and that's it. That's everything that the system allows us to have. Okay, so now what we have done now is to so all these quantities here depend on this set of parameters theta that it's quite a long, it's quite a big parameter space. Okay, so what we have done is to generate parameter values at random within a given range. Okay, and then look at for each parameter that we generated, we look whether it corresponds to this system, to this behavior, or to this behavior. Okay, and then we have done our subensembles will correspond to the high gene expression, the bistable, or to the low gene expression levels. Okay, so that's and we have generated a huge number of such parameters to have significant statistics. Okay, so once we have done that, what we need to do now is to try to find data that allows us to associate each one of these three behaviors, or at least two of them, to particular biological behavior. Okay, and to do that, we have looked at the Bernstein et al. Paper, which published in Cell in 2006. So this is quite an important paper. It's a seminal contribution because it's the one in which this Divalent Chromatin concept was introduced. Okay, and this is a very good paper, and it has data, and the data that they have comes, or at least the data that it's used for us, comes in two flavors. Okay, so the first thing that they collected is the number or the frequency of genes according to their methylation status. So what do I mean by this? So they basically counted how many genes are unmodified. So this is within a cell population, of course. So how many genes are unmodified? Okay, so there is no mark, not this one or that one. Okay, so how many genes are predominantly marked with a positive mark, and how many genes are predominantly marked with a negative mark, and how many genes are marked bivalently. So there is basically the same amount of positive and negative marks. Okay, so this is interesting to us because we can measure this thing within each one of our subensembles. Okay, within each of our subensembles, we can measure how many systems gives us an unmodified state, how many systems are dominated by the positive mark, how many systems are dominated by the negative marks, and how many systems are bivalent. Okay, so we can count these things within each one of our subensembles. And what we saw, it's interesting in the following sense. So the pattern exhibited by the bistable subensemble is basically the same as the pattern described in the experimental data corresponding to stem cells. Okay, so basically what they saw is that the frequency of unmarked, bivalent, and positively marked is of the same order, which is basically what we see here, whilst the frequency of the negatively marked genes is much lower. Okay, so it's basically this pattern that we see here. Whereas for the high gene expression, the subensemble shows the same pattern as the differentiated cells. Okay, so here the frequency of positive marks is much increased, which with respect to the bistable or with respect to the stem cell system, okay? And the frequency of the bivalent marks is considerably reduced to the same level of the frequency of the negative marks, okay? So this pattern here and this pattern here corresponds to the stem cells pattern for this one and the differentiated cells for this one, okay? For the low gene expression system, we couldn't find any sort of equivalency, okay? So that's one set of data. The other set of data that they provide is the relative expression levels of genes according to their methylation status. Okay, so again, they look at genes marked positively, genes marked negatively, and bivalent marks. Okay, for the stem cells and for the differentiated cells, and they look at the levels of the transcription expressions of those genes. Okay, and again, we can do that in our we can measure that for each one of the elements in each one of the supersamples. And what we see again is that the pattern exhibited by the transcription factor in the bistable supersample matches the experimental behavior described for the stem cells, and the same for the high and again for the high gene expression. Okay, the pattern of transcription factor expression for differentiated cells matches the pattern of expression for the within our high G expression. Okay, so with this, sorry, we can identify that so that the data supports this identification that healthy or differentiated cells that lack capability for self-renewal are identified with elements belonging to the high gene expression sample. Okay, whereas cancer or pluripotent cells that have the capability for self-renewal are identified with elements within the bistable sample. Okay, so this identification allows us to now check possible therapies against the heterogeneity of the sampling samples, okay, which allows us to evaluate if the drugs are going to be very efficient within a heterogeneous cell population or not. Okay, so and to do that, to choose targets for our in silico kind of therapy trial, if you want to call it that way, we need to analyze the samples. Okay, and particularly one thing that we have done, or we have found that it's quite useful, is comparing the marginal probability distributions associated with the specific samples. Okay, so for example, here we have the parameters corresponding to enzymes associated with modification of the K4 recipe. Okay, so this is both the enzymes that add the 3-methyl group and also the ones that remove the 3-methyl group. Okay, so that's why there are two columns here. Okay, so we can see here that, for example, if we compare the high gene expression ensemble, which again corresponds to the differentiated or the healthy cells, the ones that lack the capability for self-renewal. Okay, and so this would be the second column, and we compare it with the fourth column, which is the bistable supersample, which corresponds to the stem cell cancer cells because those are the ones that have the capability for self-renewal. Okay, we see, for example, that let's look at this case here. So, for example, here we see that there is a trend for this parameter to be lower for the differentiated cells than for the cancer cells. Okay, and the same, but the other way around here. Okay, so we have looked for these parameters that are so basically what we have done is comparing these distributions for the high GE and superensemble for the for the bistable superensemble, and identify which ones are significant, which distributions are significantly different. Okay, significantly different, but not too different, because the rationale here is that if we, for example, here, if we look at this parameter here, these two distributions are different, are significantly different. We can analyze that using the Kolmogorov-Smirnov test to compare to these two empirical distributions. So these are different, but they are not too different. So this means that the rationale to choose this instead of others is that by a slight modification, perhaps we can drive the system from the bistable to the high G state, which is the one that which is what we are interested in doing. Okay, so this is the first part, and the other part that the other thing that we have taken into account is the correlations. So basically, what we have done is to choose parameters to modify that are poorly correlated. Okay, so the rationale for this is that if the parameters are not correlated, they are sort of affecting separated parts of the system. So the idea is that if you hit these two separated parts of the system, the combination is going to be more efficient than if you hit two parameters that are strongly correlated, and possibly there isn't a synergistic effect. It's just redundant to modify those. Okay. So again, I mean so now well, that's basically the sort of description of the analysis that we do of the different examples, and we define now the target of our sort of trial. Okay, so basically what we want is to transform cancer cells. So this is in our model. These are elements of the bistable bi-ensemble. So basically, what we want to assess is what is the probability, or what is the frequency that we induce a bistable to monostable transition by changing some key parameters related to the enzymatic activity, which we choose in the way that I just described. Okay. So again, I mean, the reason for doing this is that what we want to induce is a differentiation-like process. So we want to transform a stem cell self-renewing cell into a differentiated cell that doesn't self-renew. Okay. So and what we are going to do is to change key parameters in the system to see how many of what is the proportion of the elements within the bistable subensemble that respond to this therapy by becoming monostable. So that would, in our language, that would be a sort of a differentiation inducing therapy. Okay. And what we have seen is that, so this is what these are the results of this. So basically, here is the percentage of. So basically, what we did is we took all the elements within the bistable subensemble, and we changed these parameters in a proportion given by this parameter mu. Okay, so one is no therapy. Okay, and so we increase the parameter in this direction, and we decrease the parameter in this direction. Okay, and we count how many of the elements within the bistable subensemble sort of go to a monostable state. Okay, and this is our quantification of the efficiency of the drug against the heterogeneity of the supersample. Okay, and well, these are the results. What we say is that the double parameter variations are in general more efficient than single parameter sort of treatments. Okay, well, that's not really surprising. Okay, particularly provided the way in which we have chosen things. In fact, probably the most surprising result is this one, because we see here that these single parameter variations are quite efficient. Okay, and the other thing that we did is, because these parameters are model parameters. Okay, so it's not necessarily clear how to. Also, I mean, most of these parameters are KM sort of parameters, have to do with how much substrate you need for the enzyme to start kicking in, and these parameters can be varied by changing the amount of cofactors that the enzyme needs to modify. But anyway, in any case, within our model, we can measure something that it's directly measurable in the lab, which is the activity rate of each one of the enzymes. Okay, so here we are looking at the enzyme that adds the positive mark. This is the enzyme that adds the negative mark. This is the enzyme that removes the positive mark, and this is the enzyme that removes the negative mark. Okay, and we can look at what all these variations are doing to the activity of the enzymes, which is something that in principle can be measured in the lab. And what we find is basically summarized in this slides. So basically, what we say is that the combination therapy failed to alter the median value of the activity of this enzyme that puts in the negative marks. Okay, but what it does, if you compare with the single targets, is that it sort of suppresses or sort of this high activity on ligands. Okay, so whereas it doesn't, it sort of fails to modify the general level of the enzymes, it has an effect, or a marked effect, on the high activity of ligands. Okay, and so this suggests that that cancers with high concentration or high activity of this enzyme should be more responsive to this combination therapy. The second thing that we observe is regarding the enzyme that removes the negative marks, the enzyme. Okay, and here we can see that the combination therapy does affect the level of this protein. Okay, so so it's the combination therapy leads to an increase of the activity of this enzyme, whereas it's barely affected by the single target therapies. And so this is consistent with the role of UTX as a well-known tumor suppressor in certain types of cancer. Okay, so the fact that restoring the activity levels of this protein sort of is sufficient in order to get rid of cancer. Okay. Regarding this protein, that it's the one that adds the positive mark, it basically follows the same pattern as the first one, and again the high activity outliers are the more responsive to the combination therapy. Okay, and this is essentially everything that I think I am good with time. Yes. We have formulated this model which matches the features of Divalent Chromatin. So we did quite a lot of work trying to qualitatively and quantitatively check that the model, because the model, although it's very complicated mathematically, it's very simplistic biologically. So we did quite a lot of work in trying to make sure that the model made sense biologically, and well, we think it does. The mathematical analysis, I have gingerly skipped all the details of the mathematical analysis, but this is very important. Okay, so it is very important, particularly in these machine learning days and all these cantos de sirena. It is very important to do the math, because that's going to facilitate the analysis very much. And here we have an example. By doing a lot of hard work trying to do this reduction of the dimension of the system by applying successive rounds of quasi-statistic approximations and doing it properly, we have managed to arrive to a very simple description of the system in terms of this potential function, which is everything that we need to know about the system. So this is even if you are going to do then machine learning and parameter fitting and things like that, which is obviously something quite proper to do, you should always try to do the maths first. Try to simplify the system as much as you can doing perturbation analysis or things like that. Okay, I hope I have described this parameter ensemble approach to deal with heterogeneity. I'm not sure how good a work I have done of that, but again, I mean, this is in the paper, so you can ask me if you feel curiosity about this. And then this as Javier, my experimental colleague, likes to call this exercise that we have done. He likes to call this in silico combination trials, which is quite a mouthful, but anyway. So we have found that first, I mean, by analyzing the difference between the biologically meaningful superensembles, we can get some insight onto what sort of therapy should work better. I think there is a dog complaining. And then this leads to a rationale for the selection of parameters, which in this case, we choose parameters with PDFs that are significantly different in a meaningful statistical way. And in general, we pick combinations. In general, we have observed that combinations of uncorrelated parameters do better at generating good therapies. And that's all. Thanks very much for your attention. If you have questions, I'm happy to answer.
Thank you very much for the wonderful lecture. So please, let's thank Thomas all together. So open the microphone. Questions? 1 second, and now they're like between Zoom and let's see, question. People are still. I have a very basic question. How does the dynamical system approach of slow and fast variable combine with the stochastic?
Well, it's really a stochastic version of that. So you can by doing basically the same exercise that you do with for example, in the Michaelis-Menten system. Okay, so you know that one way of getting the Michaelis-Menten approximation in this famous enzymatic reaction is by sort of non-dimensionalizing the parameters and scaling. So there is, for example, the Briggs Haldane limit, which is this one in which you have a lot of substrate and very little enzyme. Okay, so it looks like this is a proper approximation for the system. Okay, so by doing a non-dimensionalization of the time and a rescaling of the variables, you can explicitly have a system where you have a slow and fast dynamics. Okay, so here the description of your system is given in terms of a master equation. Okay, so a master equation is basically a balance between probability into a state and probability into, or probability into and probability out of a state. Okay, and you do the balance when you write an equation. Okay, so by doing the same kind of adimensionalization of your master equation, you end up with something that has explicitly scales in your system. So small parameters show up, and then you can just say, okay, so in this, if the time scale is if the time scale is very short, then the things that are evolving are the fast things, and the things that are slow are sort of frozen. Okay, and by doing that, you end up with stochastic systems that you can solve analytically. Okay, and you can find the probability distribution, the adiabatic limit of the probability density function. Okay, and with that you can average. So knowing that, you can do an average, and you go to the next level. And in the next level, you do this.
So you do fast as low variable on the
master
equation,
or the Fokker-Planck equation. I mean,
yes.
Here the description was the master equation, but if you have something like a diffusion approximation in terms of a Fokker-Planck equation, it's exactly.
And then okay, very nice. I think I will ask you, Joseph, to give us mini course about this. Just about this, that would be very interesting. Very, very nice. Other question? We have already, people are a little bit shy in the after lunch here. Okay, so I guess enough for the moment. But thank you very much for the great talk. It would be possible to have the slides, because we would like.
Yes, I will send them.
Yeah, we would like to put them in the conference webpage so people can, because they're.
A PDF is fine, or do you prefer?
I think the PDF is perfect. Yes, it's perfect.
Perhaps I will send you a Dropbox link or something like that, so I will fill you.
Yeah, because every talk is very different from the other, and I think people, this is the idea, is to cross people from one area to kind of be exposed to others, and so people have to kind of review with more, time, different techniques. Thank you very much. Great talk.
Thank you.
As always. Thank you.
Bye. Cheers.
Cheers. Bye-bye. So I pass the word to Fabrizio or Adriano for the next speaker.
Okay.
Yes, Adriano.
So let's continue here. Now we are going to have André Fujita. Thank you very much, André, for accepting to give us a talk, to present a little bit of your work for us. So André, I'm going to say some words about André. André Fujita obtained his bachelor in computer science and PhD in bioinformatics from the University of São Paulo. He was a postdoc at the Human Genome Center at the University of Tokyo, and the computer science research program of Hiken in Japan. He was visiting research at the Cancer Research Institute of the University of Kanazawa, and guest researcher at Leipzig. He also received several national and international awards, such as the Alexander von Humboldt Fellowship in Germany and the Newton Advanced Fellowship by the Royal Society. and the young research highlighted by the Brazilian Association for Bioinformatics and Computational Biology. His interest in things like associated with connectivity and network analysis, statistical analysis, both with application in biological data analysis. And currently, he is an associate professor at the Department of Computer Science of the Institute of Mathematics and Statistics of the University of São Paulo, and he's the vice coordinator of the graduate program in bioinformatics. Once again, thank you very much, Andrea, for your time to be here.
Thank you very much for this opportunity to present our work here. I'm trying to share my slides. Let's see it works. Can you see my slides?
Yeah, I'm seeing it. Everyone in the audience is also seeing?
Yes.
Okay.
Okay. Thank you very much. Thank you very much, Adriano. Okay, today I want to talk a little bit about network statistics. It's something that we are working in our group for the last 10 years or a little bit more. I will show some several techniques to analyze networks statistically. So I mean, several tools that you have already seen in basic statistics like correlation, t-test, analysis of variance. I will show the equivalent methods for networks. Okay. So, well, as the title describes, the object of our study is a graph, and the first graph in the history. Well, maybe before it, there were some examples of graphs, but what is very well known is the graph modeled by Euler in 1736 to solve the problem of the 7 bridges of Königsberg. You can see here 4 places, 4 portions of land, A, B, C, and D, and 7 bridges associated here with this green line. And at that moment, there was a kind of problem that people try to solve. Is it possible to travel from by all the bridges exactly only once? Several people tried at that time, but no one solved this problem until Euler proved that this problem there was no solution. So it was a negative result that it was impossible to travel by all the bridges only once. And he modeled this problem as the first graph in the history. He showed that each node is described by the circle and the bridges by edges here. And this probably, and it was nowadays it's known as the first graph. And then I jump from the 18th century to the 20th century, that we have the beginning of the area of random graphs. It started with Paul Erdős and Alfred Rényi, that they proposed the first random graph of the history. Very briefly, it's composed a graph. You have n nodes that is represented by these dots and edges represented by these lines. And they said that, well, given a probability P, you connect a pair of vertices. For example, with P equals to 0, you have no edges. With P equals to 0.1, you connect with probability 0.1 each pair of vertices, okay? So it's very easy to see that if I give you a P equals to 0, you obtain an empty graph, and if P equals to 1, you obtain a complete graph. Okay, so you obtain several random graphs. Oh, sorry, it's not full screen. How can I put it full screen? Sorry, I didn't notice before. Now it's better. Sorry. And since then, several other random graph models were presented, like random plane networks, stochastic block models, etc., etc., etc. Until at the end of the 20th century, in 1998 and 1999, probably you have already heard about these 2 random graph models, I mean complex network models. One was proposed by Watson and Strogatz in 1998. And the second one that is very, very famous is the Barbaz-Albert random complex network model published in Science. Very briefly, the Watson-Strogatz complex network model consists of a regular graph that you, for each vertex, you connect to its neighbors. and you give them the number of edges. And then with probability P, you shuffle these edges. So if P equals to 0, you have this regular graph here, a circular graph. And if P equals to 1, you obtain a complete random graph, like edge in. But if P is between 0 and 1, you obtain this kind of graph. And especially if P is very close to 0, like 0.1 or 0.05, for example, you obtain this structure of graph. It's very useful to model social networks. Okay, because you have some clusters, you can imagine that each vertex here is a person in a social network, and the edges represent their relationship, for example, friendship. So you obtain some clusters that are people that are very close, and that they are also connected to people that are very far, for example, here. Okay, so it was very useful to model social networks. These structures are different than you obtain by an edge-rendered random graph model. And the second one is the Barbaz-Albert model. You start with a seed graph, very small graph, for example, 2, 3, 4, or 5 nodes, and you connect a new vertex proportionally to the degree of the existing vertex in the graph. For example, here, this red node here is the new one, and what's the probability to connect to each vertex already existing on the graph? So this one has the highest degree. So the probability that this red one connects to this black node here is higher than the others. Okay, and then you continue this process until you obtain the desired number of vertices. And finally, what you obtain is a graph that has the following structure. You have very few nodes with very high degree, and a lot of many nodes with very low degree. Okay, and it's very useful, for example, to model metabolic networks or protein-protein interaction networks, several biological networks, because we know that metabolic networks, you have several metabolites that present very high degree, and many metabolites that present very low degree. And since then, at the beginning of the 21st century, people started to use this Barbaz-Albert model or Watt-Strogatz model to model several, not only limited to biological, but also social networks or financial networks, etc. And people start to analyze networks structurally. For example, given 2 proteins or 2 nodes or 2 metabolites, for example, what is the shortest path between them? Or can we identify number of triangles? That means how the nodes are clustered, or something like a modulus in the network, like these 2. Or also which nodes are very important in your network. For example, here we can see that this black node here maybe is very important because, well, if you knock down this node, this vertex, probably you split this network into 2 subnetworks, the left one and the right one. Okay, but of course, depending on the definition of importance, maybe this red node may also be very important because it presents a very high degree. Okay, so the importance of the nodes can be defined in many ways, depending on which kind of interpretation or you are designing. People also start to analyze motifs that are very small structures, small patterns that you can find in your network. And in 2010, more about 2010, what happened is that people start to analyze networks in medicine, especially diseasome, I mean the network of disease, or networks, gene regulatory networks, functional brain networks, etc. I mean biological networks in general that can describe several diseases or mental disorders. But here, what happens is that we have a very critical problem because, well, you know that we have variability. For example, my gene regulatory network is not exactly the same as yours. My functional brain network is not exactly the same if I compare mine with yours. So why? Because, well, we are unique. We have this variability. We have randomness in data. Okay, so it's not very reasonable to compare networks. I mean, mathematically or in terms of computer science, like I think it's not trying to find isomorphism, like what computer scientists and mathematicians like. It's not good enough in biological data analysis. And so what may be the alternative approach? I think the statistics is a very reasonable approach, because statistics you deal with randomness, fluctuation data. And in fact, Rubinov and Spons, they publish in the journal Neuroimaging. Neuroimaging is one of the top neuroimaging journals. They said that brain networks will require the development of accurate statistical comparison tools. So, in other words, how can we compare functional brain networks statistically and not in the sense of isomorphism? And also, John Hopcroft is a Turing awardee. He also said that discrete mathematics was a big part of computer science, but in the future, it's probably going to move to be probability and statistics. And well, in fact, we see it nowadays when we analyze, for example, deep learning or machine learning. We can see that probability and statistics are dominating computer science. So let's see a little bit about the statistics. The first wave of the modern statistics, because we can, statistics we can, we have several reports saying that in Greek we have some ideas of mean or mode or median, but here I am calling as the statistics as what we know as the modern statistics. For example, the concept of correlation, or principal component analysis, or chi-square test, okay, that they were developed by Francis Galton and Carl Pearson. For example, correlation, Pearson correlation was developed by Galton and Pearson. Principal component analysis that probably many of you use was developed by Pearson, chi-square test, etc. And the second wave came with William Gosset. William Gosset, some of you may not know him. He developed the t-test, or his nickname was, pseudonym was the student test. And Ronald Fisher, well, the father of multivariate statistics with discriminant analysis, analysis of variance, ANOVA, maximum likelihood, etc. And the third wave came with Jerzy Neyman, with the idea of confidence interval, and Egon Pearson with the Neyman-Pearson lemma. And not a coincidence of his surname, Egon Pearson is son of Carl Pearson. And interestingly, before one of the most popular papers of William Gosset, the t-test, it was published 1 year after he stayed 1 or 2 years in internship at Carl Pearson's lab at UCL. Okay, so well, you can see here a very small cluster of statisticians working together in the UK. And there are many other very well-known statisticians like Clive Granger at the end of 20th century. He's very well known with the concept of Granger causality between time series, but he won the Nobel Prize with the concept of co-integration. Hirotsuga Kaiki is one of the scientists that introduced the concept of model selection in statistics, and so on and so forth. And what I want to show you are these methods that probably many of you already seen in basic statistics like correlation, PCA, chi-square test. And how can we do it on graphs? But the problem is, in graph theory, we studied this kind of object, I mean the graph. It's a mathematical object composed of nodes and edges. And in statistical theory, we analyze numbers, essentially numbers, okay? So how can we combine these two theories to obtain a statistical approach on graphs? This is the main problem, okay? So what we do is, given a graph, for example, this small one, we can translate this graph into a adjacency matrix. So if you have, for example, you put the nodes on the columns and rows here, and you put 0 if the two nodes are not connected, and 1 if two nodes are connected. For example, between 1 and 2, there is no edge here, so they are not connected. So row 1, column 2, you put 0, and row 2, column 1, you put 0. Between 1 and 3, you have an edge. So row 1, column 3, you put on 1, and row 3, column 1, you put 1, and so on and so forth, and you obtain this symmetric matrix. By simple linear algebra, you can decompose this matrix into eigenvalues and eigenvectors, and we call the set of eigenvalues as our spectrum of this graph, okay? And so if you have a graph composed of any verts, for example, 1000, you obtain a matrix composed of 1000 rows and 1000 columns, and you obtain 1000 eigenvalues. So you can construct a spectral distribution of eigenvalues, what we call as the, and then what's the interesting part of the spectral distribution is that given if two graphs present different spectral distributions, like here in center and in the right graph, you can say that these two graphs were generated by different stochastic process. But if the two spectral distributions are not different, I mean they are equal, you cannot say anything. Maybe the graphs were generated by different stochastic processes, because we have some very rare examples like co-spectral graphs, but usually they were generated by the same stochastic process, but it's not true every time. So, but at least we know that if two spectral distributions are different, we know that they were generated by different stochastic processes, okay? And the study of the spectrum of the graph is very well known since the 19th century with the concept of eigenvector centrality, that is the basic of the PageRank used by Google, graph energy that were first published in chemistry, and several other properties like the largest eigenvalue, second largest eigenvalue, etc., etc., etc. Okay, so it's a very well-known field in maths. Okay. So what we use is spectral distribution of the graph. And first, what we did was just apply the concept of channel entropy on the spectral distribution, and we defined something that we called as the graph spectral entropy. It's this one. It's essentially the application of the channel entropy here. Okay. And interestingly, what we obtained is intuition of the spectral entropy is that, for example, for the Edison-Renyi random graph model, that we have a set of vertices and a probability P that you connect a pair of vertices. If P equals to 0, you obtain an empty graph, and for P equals to 1, you obtain a complete graph. And in these two cases, you obtain the lowest entropy here. Okay, so, and these two graphs are the most predictable graphs, because if I ask you if a pair of vertices are connected or not, you will of course say that they are not connected here for P equals to 0, and in this case for P equals to 1, you will say that they are connected. So it's totally predictable. And which graph is the most unpredictable one? It's for P equals to 0.5 here. That in fact you obtain the highest entropy here. Okay, because if I ask you if a pair of vertices are connected, you probably obtain a correct answer only half of the times, on average. Another example is what's the Stroker's random graph model that I already showed you. For P equals to 0, you obtain a regular graph, so it's totally predictable, so you obtain a very low entropy, and as you increase the probability P, I mean the randomness of the graph, you obtain higher entropies, okay? And there are several other examples that I will skip the slide here. And I show you two applications of the graph spectral entropy. One is both of them in functional brain networks. We analyzed hundreds of, about 1,000 children, more or less half of them controls and another half with ADHD here, and here for more or less 500 children with autism. And what we construct the functional brain networks, so you obtain a graph, a brain graph. We cluster the nodes, and then we obtain the 4 clusters here that we can, they are represented by these 4 colors, red, blue, green, and yellow. And what we compared which subgraph here presents a different graph spectral entropy between controls and ADHD here, and controls versus autism here. And what we obtained is that this green area, majorly composed by the somatomotor region, and here by the cerebellum, this orange one, present the highest, a higher graph spectral entropy if compared to controls. And in both cases, we can, here, the cerebellum is a very well-known area related to autism, and here we obtain other areas that were not described yet in ADHD, but were belong to this green area. And now, finally, I will show you several methods, statistical methods on graphs that are based on the graph spectral entropy. The first one is the parameter estimator. Well, given a set of data, for example, ages or IQ score in physics, etc., and given a model, for example, a normal distribution, a model you have a set of parameters. For example, for the normal distribution, you have the mean mu and the variance sigma square. Okay. Well, given a set of data, a data set and a model, the first thing that we usually do is to estimate the parameters of the model. For example, in this case, we have maximum likelihood estimators for the mean and the variance sigma square. For example, for the mean, we can maximum likelihood estimator is just to sum all the data points and divide by n. So you'll be taking x bar. That's a very good estimator for mu. And a Bayes estimator that's, well, you subtract the average, the mean from each data point, square it, and then divide by n. And then you'll be taking a Bayes estimator for maximum likelihood estimator for the variance here. Okay. Well, and for graphs, given this graph, I want to estimate the probability P that I used here to connect the graphs. For the edge-linear random graph model, it's very easy because, intuitively, you need just to count how many edges you have in the graph and divide by total number of possible edges. For example, if you have n nodes, it's n2 by 2, which is the number of possible edges. And here I counted 92 edges. So I obtain here 0.51. That's very close to the parameter that I used here. In fact, that is 0.50. So this is the how we can estimate the parameter P for the edge-linear random graph. But the problem is for more complex networks. For example, Watt-Strugatz. You have a regular graph, and with probability P, you shuffle the edges here. So given this kind of graph, how can you estimate the probability P that I use to generate this graph? This is the question. It's a very difficult problem. I don't know any kind of analytical formula to estimate this probability P. And what we propose is, well, we can do something very similar to the maximum likelihood estimator. Instead of using likelihood function that we don't know for graphs, we can replace it by the spectral distribution. So suppose that you have an observed graph, for example, your gene regulatory network and a model, and I would like to estimate the parameters of this model. So I compare the spectral distribution of the observed graph with the spectral distribution of the model. And here I try several parameters theta here, and I select one that minimizes the distance between the observed graph spectral distribution and the model spectral distribution. And the parameter theta here that minimizes this distance is our estimated parameter. And this distance, what we use is the Kullback-Leibler divergence measure. Okay. These are some examples that show, for example, we have the edge-linear random graph model, geometric, Barbaz-Albert, Watt-Strugatz, K-regular graph, etc. Between parentheses, we have the user parameter, and here the size of the graph. And you can see here that as we increase the graph size, you obtain more accurate parameters here that are very close to the ones described between parentheses. Okay, so it seems that it in fact works. But the problem is that this method is highly, the computational cost is very high. We can do it for graphs composed of a few hundred nodes, up to 1,000 to 2,000, I think is reasonable. But it's impossible for graphs composed of 1 million or 2 million nodes. Okay. So what we proposed here is, well, one PhD student, he developed a method to estimate this. One of the bottlenecks is the estimation of the spectral distribution, because if you're a computer scientist, you know that it's n cubic. N is the number of nodes of the graph, so the computational cost is very, very high. So what he did was to develop a very efficient approximation for the spectral distribution that's based only on the degrees of the graph. It works only for locally tree-like networks, I mean very, very sparse networks. But in practice, it's not a problem, because we know that social networks, biological networks, several empirical networks are in fact sparse, so it's not a problem. And he can do it in linear space and time, so it's extremely fast. And we replaced the Koba-Kleiber divergence by the L1 norm. Why? Because another PhD student showed that if we she proved that if we replace the Koba-Kleiber divergence by the L1 norm, we can prove that our estimator here is consistent. So, and here we have an optimization problem that we were using a grid search, and then we replaced it by a ternary search, assuming that the L1 norm here is concave. Okay. So what we obtained is a very fast parameter estimator. Just for illustration, we simulate a Barbaz-Albert random graph model composed of 5 million nodes. And if we use our previous method, we estimated that it needed approximately 900 years to estimate parameter. And in this case, we used only 2 days, only 48 hours on average. Okay, so well, now we can estimate parameters for very, very large graphs, like orders of millions. Okay. A second approach is model selection. Suppose you have a data set, and I propose you several models, for example, 3 models, a very complex model that you link all the data points, a simple model that you cannot obtain this sine cosine curve, and an appropriate model here. So the complex model, you can see that it's very easy to see that it's overfitted data. The simple model cannot identify this shape, this sine cosine shape. And appropriate model here in the center seems to be more or less good. So the question is, how can we select the best model? Okay, this is the problem of the model selection approach. Well, Hirosuga Kaiki, in 1974, he proposed the Kaiki information criterion. It's a criterion to select a model, given a list of models. So for random graphs, what we did is give an observed graph, for example, a metabolic network, and 3 reference models, for example, model 1, model 2, model 3. I would like to check which model is the best one, is the one that best fits my data. Okay. So the idea again is very similar to the Kaiki. Again, we use the same trick. Instead of using likelihood function that we don't know for random graph models, we replace it by the spectral distribution of the graph and penalize it by twice the number of parameters of the model, just to avoid overfit like a Kaiki. And we'll be taking the model selection approach for graphs. Okay. It doesn't mean that the select model is the one that nature or God used to construct your empirical graph, but it just means that this model is the one that best fits the data. Just one illustration. We select 7 or 8 protein-protein interaction networks for several species. Our list of random graph models are these 3, RGRNE, scale-free, and small world. We used our approach, and we note that for all of them, the scale-free graph was the selected one. It means that it's among these 3, the scale-free network is the one that best fits these protein-protein interaction networks, and it corroborates the data that we already know by the literature. Okay, but it doesn't mean that God or nature created protein-protein interaction networks using scale-free model. It just means that among these 3 is the one that best fits. Maybe in 10 years on someone comes with another model that better fits protein-protein interaction networks than scale-free. Okay, it's like in physics that we know that some centuries ago, the Newton model was the one that best fitted data, our nature. But then in the 20th century, Albert Einstein showed that, well, if your object is moving very close to light speed, or very extreme case, Newton's model, Einstein model fit better the data. Okay. Comparison test, t-test, analysis of variance, etc. Well, William Goss at the beginning of the 19th century show a test that, given two data sets, for example, men's high and women high, we want to compare if the heights are equal or not, for example. So you can, well, taking into account the variability of the data. So usually what we do is a t-test. We would like to compare the means considering the variance of the data. Okay, this is a t-test. And for graphs, suppose that you have a set of graphs, gene regulated networks, for example, healthy people, and a set of gene regulated networks of people diagnosed with diabetes, for example. And I want you to test if they are equal or not in some sense. And here the concept equal means if they were generated by the same model, okay? Because it's nonsense to compare two networks, I mean, in a pairwise manner, because of course they will not be exactly equal. Because even if you compare two people that are healthy, I mean, belong to the same group, they are not exactly equal. Okay, so because they are fluctuation or randomness. So what we did is it translated each graph into its, we obtained its spectral distribution, and we compared the spectral distribution. I mean, the average of the spectral distributions. Okay, this is the t-test for graphs. And once we have the t-test, it's very reasonable to think that we can generalize it for k-tests, for k groups, like the analysis of variance, that instead of comparing two groups, we want to compare 3 or 4 or 5 groups simultaneously. And then the new hypothesis here are the means are equal, or otherwise alternative hypothesis, at least one of the means is not equal. Okay. And for graphs, the idea is exactly the same. Instead of two groups, we have k groups of graphs, for example, controls, people diagnosed with Asperger, and people diagnosed with autism. And I would like to test if the functional brain networks of these 3 groups are equal or not, or if at least one of them are different. Okay. The generalization is very straightforward. It's very similar to the analysis of variance of Fisher, proposed by Fisher, that we compare if the average of spectral distributions to the average of averages of all spectral distributions. Because if one of them are different from the others, probably the distance between the specific one to the average of the average will be large. Okay, so this is the idea. We applied it into functional brain networks. Again, we compared controls versus Asperger versus autism. What we obtained is that the cerebellum again is different, and then we performed a pairwise comparison. I mean, controls versus Asperger, control versus autism, and Asperger versus autism. And what we obtained is that controls versus autism is a pair that makes the difference statistically significant here. Okay. But then you can ask me, okay, Andre, but in the real world, we don't have sets of networks. We usually have only one observation of the network. For example, the protein-protein interaction network, we usually have only one protein-protein interaction network for each species, or one metabolic network for each species, or one social network. For example, we have a Facebook network of Brazil and a Facebook network of Italy, or Spain, or Germany, or Japan, China. So how can we compare networks when we do not have replicates? At a first glance, it seems to be impossible because we don't have replicates. But if we know the model, we can do it because then we can compare the parameters of the graphs if they are equal or not. So what we do here is to estimate the parameter of each graph and then compare if the parameters of each of the graphs are equal or not. And how can we do it? The first step is, if you check here, you will see that these 5 steps are exactly the same as done by Fisher in the analysis of variance. First, you estimate the parameters of the graphs, and you can use our Koba-Kleiber divergence-based estimator describing previously. then you can estimate the variance of the parameter using a parametric bootstrap. And then steps 3, 4, 5 are exactly the same as developed by Fisher. And you obtain analysis of variance for graphs, for single graphs, okay? This is just an example that we compare the protein-protein interaction networks of 6, I think, yes, I think 6 bacterial species. And here are the p-values that we can see that for some of them, in a pairwise manner, we obtain significant p-values. Yes, they were significantly different. And now the concept of correlation. Okay, Galton and Pearson proposed in the 1880s the concept of Galton-Pearson correlation, the Pearson correlation. Suppose that you have two data sets here, X and Y, for example, scoring physics and scoring maths, for example. And we would like to see if they are dependent or not, linearly dependent or not. So you can put the score in maths here in the x-axis and the score in physics on y-axis. So you obtain this scatter plot, and you can compute the linear regression here and calculate the angular coefficient here. And you essentially obtain the Pearson correlation. So here it's very easy to see that we have clearly a correlated data between X and Y are highly correlated here, okay? So if I give you the value of X, it's a very good hint to guess the value of Y, okay? For example, if I tell you that my score in maths was, for example, 6, probably you will try and I ask you my score in physics, you will say, okay, if you got 6 in maths, probably you got something very similar to 6 on physics too. Okay, it's very rare that someone that obtained 8 in maths, you obtain 2 in physics. Okay, so data are very dependent. Okay, and for graphs, given 2 sets of graphs, for example, the function, the gene regulatory network of my blood and the gene regulatory network of my lung, for example. And I want to see if they're correlated or not. Just observing these 2 data sets, very difficult to say that they are correlated or not. But now, if I tell you that there is a model behind these graphs that were used to construct these graphs and a set of parameters, we can define a correlation between graphs as if those parameters of the graphs are correlated, the graphs are correlated, okay? So if you know the parameter of graph X and we know the parameters of graph Y, and here clearly they are correlated, we can say that these 2 sets of graphs are correlated. But in general, we don't know the model of the graph. So what we observe is that usually for many random graph models, the largest eigenvalue of the adjacent matrix is a function of the random graph model parameter, okay? So what we use, in fact, is the largest eigenvalue as a proxy of the parameter of the graph. And we can see here that, in fact, the spectral radius, I mean, the largest eigenvalue, is monotonically correlated to the parameter of the graph for this, at least for these 5 random graph models, okay? So it's a very good proxy for it. Here's just application that we compare the functional brain networks correlation between control area, default modes, motor, and visual. And we observe that correlation between these 3 pairs here is higher in autism than in controls. Granger causality. Another concept is Granger causality. If you have given 2 time series, and I can predict the present and future of one time series using past values of another time series, for example, if I can predict Y, future values of Y using past values of X, I can say that X Granger causes Y, okay? This is the concept of Granger causality. Very roughly speaking, it's the concept of Granger causality. And now suppose that we have a time series, we have time component here, so you can imagine that the graphs are changing over time. And here, they are not correlated here, the parameters of the graph, but they're correlated in lag. For example, if I analyze time point 0, it's highly correlated to Y graph time point 1 and so on and so forth. So in this case, I can say that graph X Granger calls graph Y. Okay. This is just a simple application that we show how is the causality between right and left hemisphere between people, healthy people and people diagnosed with autism. And we obtain that these two regions here, Granger calls the right hemisphere regions, Granger calls left hemisphere of the brain. In all autism more than in healthy people. Unsupervised classification. One of the simple examples of unsupervised classification is clustering. Usually when we talk about graph clustering, we imagine something like this. We usually cluster the nodes, very highly connected nodes into clusters and very sparsely connected nodes into different clusters. But here our problem consists of clustering the graphs themselves instead of the nodes of the graphs. So I want to cluster if graphs, similar graphs into the same cluster and very different graphs into different clusters. Okay, so the problem is totally different from the B panel here. So what we use is very similar to the classical mixture of Gaussian distributions, mixture models. Let's suppose that you have a data set and we assume that you have a number K of clusters. And we assume that each cluster K is a Gaussian distribution. For example, it could be another distribution like a T distribution, chi-square distribution, anyone. Here we usually we assume that they are Gaussian distributions. And we verify which the probability each item, which the probability to come from each Gaussian distribution, and we select the highest one to say that this specific item came from the specific Gaussian distribution. Okay. For graphs, we do exactly the same. Instead of assuming Gaussian distributions, we assume that they came from random graph models, K random graph models, and we do the same steps of the mixture of Gaussian distributions. And one application is in functional brain networks of monkeys. Chibi1 and Chibi2 are the same monkey called Chibi in 2 different days. The monkey is awakened, and at some point here in this green line, it's injected a drug, anesthesia. So the monkey is anesthetized in this at this point here, and this blue solid line is the moment that the medical doctor says that the monkey is sleeping. He's a total anesthetized and he lost his conscious. And we collect the brain signal of the monkey and construct functional brain networks for each second, and we cluster the functional brain networks. So we obtain the unsupervised clustering here that we can see that this dashed line is the point that we have a phase transition, that the monkey changes his state from awake to anesthetized at this point. That's after the injection of the drug and before the medical doctor, in fact, evaluated that the monkey lost his conscious. And here's the opposite side, that the monkey was anesthetized and then he started to awake, and we could predict it before the medical doctor. Okay. And the last method is the supervised classification. The classical supervised classification approach is the linear discriminant analysis proposed by Fisher. Of course, you have several others like sport vector machines or random forest, etc. So we base it here on linear discriminant analysis. You have a data set and we project it into a lower space and we obtain a classifier. For example, in this case, we set the classifier more or less here, the threshold, and we say that upper this threshold is circles and below this threshold are squares, for example. We do the same for graphs. Instead of using the graph itself, we use the spectral distribution and develop the convolution-based linear discriminant analysis. So we use the convolution approach. So we replace all the dot products of the Fisher linear discriminant analysis by convolutions. And we obtain exactly the same linear discrimination analysis of Fisher. But for at that time, we submitted the paper and reviewers said, okay, but what you have is not a linear discriminant analysis for graphs, but you have a linear discriminant analysis for a more general problem that's for functions. we said yes. In fact, well, we agreed with the reviewer. We rewrote the paper, the entire introduction, and we showed we present the paper with after one review as a linear discriminant analysis for functions. This application that we show that our approach here are the CLDA and WCLDA that are our proposal methods that present average error rate lower than the other competitive approach. And one of the biggest advantage of our approach is that it's not only the classification error, but also the interpretation, because we can identify which frequency of the signal is the one that's most important for classification. For example, here we have two cardiac signals, electroactivity of the heart, and in panel B we have for many people, okay. And here we have the electroactivity, the filter that our approach obtained. Here's the electroactivity of the heart obtained after field application of filter. And here you can obtain which frequency is the most important to discriminate people that have a heart disease and healthy people. So we can do a frequency analysis. Well, it's very useful, for example, in the brain, because in the brain, well, when you analyze EEG, for example, electrocardiography or encephalography, it's very difficult to filter out information from heartbeat or respiration, for example. So, but you know the frequency of heartbeat. You know that's more or less between 60 to 80 heartbeats per minute. So in this case, you can use this kind of filter and isolate this effect, okay? All of these methods are already implemented in R, and you can obtain it from this link. It's totally free, open. I like to thank all my current and former students, postdocs, collaborators, and the funding agencies. Thank you very much for your attention. And this is my email address, so please feel free to contact me. Okay, thank you very much.
Okay, let's open our microphone and congratulate Professor André Fujita. Thank you very much, André, for a very nice talk, very nice survey of your work, your new tools, your developed tools in graphs, statistical analysis of graphs. Very, very interesting. Are there any questions from the audience? Okay, no questions. Okay. I do not have a specific question to you, but I can say that I'm very eager now to take a closer look in your work. It was a very good teaser of your work. So I guess also Professor Stefanella also is very interested in to see more closer your work, take a look better in your work. She's working in complex networks for a while, so for sure she wants to talk to you later on. But right now I guess she's busy with some classy duties, but she already asked us to ask for you later if you could share your slides, if it's going to be very helpful for the classes later on. Okay.
Thank you.
Thank you very much, André. Very nice talk. Okay, let's congratulate André again.
Thank you.
I guess it's, yeah. Okay, now Neil, are you set? Oh, okay.
Hello.
Hello.
So should I go ahead and share screen?
Okay, yeah. I will introduce you. Okay, so many thanks, Neil, for the opportunity to listen to you, to be here with us, to give us this talk. So Neil, Professor Neil, actually, Professor Neil Maga, she obtained his PhD in system biology from Harvard University in 2013. After that, she passed through different and very ranked important American universities like the MIT, Brown University, and University of Washington, where I guess she worked with professors Nathan Coote and Steve Brunton. She's an assistant professor at the Engineering Science and Applied Mathematics Department in the Northwestern University in Illinois. She's also a preceptor for interdisciplinary biological science. She's a member of the NSF Simon Center for Quantitative Biology, member of the Center for Synthetic Biology, and affiliated of the Center for Engineering Sustainability and Resilience. The phrase in his page is top-down. Her research interest is top-down data-driven methods meets bottom-up mechanistic modeling. I guess this is all about what we are going to hear from her today. So thank you very much, Neil, once again. You have the word.
Thank you very much, Adriano, for inviting me. I am very happy to share my work today, and I'm also very happy that you were willing to let me do this remote rather than in person, because it's allowing me to do this despite being a little bit sick. Okay, so today I want to tell you a couple different stories. Before I say that, before I get started, I want to say if anyone has any interest in the methods, especially since I can't be there in person to hang out and answer questions afterwards, please do email me, and I would be happy, especially with students or postdocs or trainees, to set up a time to chat. Okay, so I'm going to tell you a couple different stories about data-driven model construction. And the first, before I get started, I want to just thank my group. This is my group at Northwestern. Most of the work from my group that I'll be talking about today is from Helena, Sasha, and Julie. And then I'll also be talking about a little bit of work from some collaborators. As you can see, my group works on a broad variety of different things, but at the core, we're trying to develop mathematical models to describe different systems to understand them better and develop methods to build those mathematical models faster. So within biology, some of the biological questions that I've been motivated are really about metabolism, and especially the spatial temporal organization of metabolism within bacterial cells. So I'm interested in how co-localizing enzymes together or with putting them inside of compartments, or just putting them at the membrane, perhaps near a transporter, how those different types of spatial organizations can impact flux through the network. And especially questions like which types of organization would work best for different reaction pathways. This interest comes out of working on a variety of natural systems, including this is the propane diol utilization system in Salmonella, but it also comes from the idea of being able to engineer these type of systems to produce new products that we would be interested in. So biofuels, energetics, drugs, and my collaborators, some of my collaborators at Northwestern in the Center for Synthetic Biology are actively working on building these systems. As they're trying to build these systems, though, a lot of problems come up. So you try to put a new system inside, for example, a protein microcompartment. So it's like a protein capsid that can hold the enzymes and prevent the metabolites from getting in and out as easily as they would without the barrier. You have all sorts of problems. So sometimes you put a pathway in and it just doesn't run the way you were expecting to. Is that because there's competing reactions? or are you missing some cofactors that are limiting flux? You know, a big question that comes up is just what is the rate limiting step within these systems? And that can vary widely depending on what the experimental conditions of the system are. And so mathematical modeling can be one way to address these questions and really identify where the rate limiting steps in these metabolic networks are. But in some ways, especially with high throughput methods for measuring the concentrations of these metabolites over time, the modelers, which I think of myself as a modeler, are starting to have trouble keeping up with the amount of data that's produced and just the number of experimental conditions under which we need to be able to describe the system. So what my group does is tries to develop new methods to build models very quickly. And we're big proponents of a framework that looks something like this here on the left. The idea being that we would pose many possible models, which, you know, going back to the last slide, could essentially be representing many different rate limiting steps or many different cofactors coming in and or being missing and limiting what's going on. We have some data from the experiments, especially from some of these high throughput robotics experiments. And then we perform some amount of model selection to down select a set of models. We would like to then be able to characterize the certainty of the models, meaning between the models, and also the parameters within those models so that we can iteratively suggest new experiments to further down select those models. So this is kind of the overall picture of what we do in the group. And I'm going to talk you through 3 different main points. So first, I'm going to go through a framework which my collaborators have named GAMES, which is a framework for systematic model development. And this is actually more of what I think of as a traditional model development framework, but it has some really nice checks and tools in it that you know, even someone who has maybe been developing models and is familiar with model development may find useful. So I also think it would be very useful for anybody who is maybe has more biological training, but has gotten comfortable with building ordinary differential equation models and systems biology type models, and then simulating those models, and really wants a rigorous way of testing whether their model is robust. So then I'll also talk about this, some background on sparse model selection as an approach to overcome the complexity of all of the possible things that could be going on in the system. And finally, I'll talk about some recent work on using sparse model selection, but in the context where you can't actually measure all of the important state variables that are in your system. Okay, so first, this GAMES framework. So GAMES is a dynamic model development workflow to characterize synthetic genetic systems. And technically the methods that are involved in this workflow, so I should say most of the basic methods, these things were already available in the literature. What Kate, who's the first author on this paper, has done is really put them together into a nice systematic framework. She also has an absolutely wonderful code base. So if you're getting started in trying to build, evaluate, and test your models with data, then I would highly suggest looking at her code base as a potential way to not have to build all of your parameter estimation tools and evaluation tools from scratch. But what this workflow does is it basically goes through the steps of formulating the model and collecting the data, evaluating whether the parameter estimation technique you're going to use works for that particular model, doing the actual parameter estimation, so fitting the model to training data, assessing whether that model fit is robust and unique, so assessing whether the parameters are actually identifiable given that the model you have, and then comparing between candidate models. And this is not just a linear workflow. Depending on the results you get out of each of those steps, you may have to go back and reformulate. So again, if you're somebody who has developed mathematical models to model biological systems, this is not anything new. It just brings together in a rigorous way the approach, and at least Kate, Joe, Netta, and Josh and I thought, if you are developing a model, you go through these steps and often you only show the final product, and actually acknowledging what the different steps are, and potentially rather than just showing the final product and its fit to the data, it could be useful to have kind of these checkpoints that you show during model development to show how the model came about. Okay, so the other piece of this is that the whole thing is couched in this, example, which is a very systems biology, synthetic biology example. It's a transcription factor model. And so if you are familiar with that type of system, this will be a nice example that is fairly followable. Okay, so GAMES, just the backronym that Kate and I think probably Josh came up with, was a workflow for generation and analysis of models for exploring synthetic systems. And again, this is going to be really useful for those who are already comfortable with ordinary differential equations models and some programming, but maybe need some guidance in how to evaluate their model and how to fit to data. And I should say, I actually suggested this code base in this paper to one of my postdocs, who is, a very well-trained mathematician, but just didn't have very much experience with doing parameter estimation and working with data. So I think it's not only useful for people who are maybe coming from a more biological background into a modeling, trying to do modeling, but also potentially for people who are from a more mathematical background, know some biology or have learned some biology, but don't necessarily have experience with working with data and model fitting. Okay, so here is the case study. And this is basically, again, as I said, it's a transcription factor model, but the transcription factor has 3 different components: a DNA binding domain, a ligand, and a activation domain. And so if only the DNA binding domain is bound, then it actually inhibits transcription. Whereas if all 3 components are bound together, then you actually get active transcription of your reporter. So it's a very simple model, or a very simple system that we're using as a case study. And basically what we do is we have a model of this system, which we use as ground truth. And then we produce data from that. And then we pretend that data is real data, and we go through our model construction process or model testing process as if we didn't know that was the ground truth. All right. So just to give you an idea, or to show what this type of model looks like, these are ordinary differential equations that describe the time-dependent trajectories of each state variable in the system. And so there's sort of these standard rate of change of the different components is the production of those components and the degradation of those components. So for example, here's our DNA binding domain. It's got a transcription rate and then a degradation rate that is dependent on the concentration of the DNA binding domain. You can write down the full set of equations, and for this case study, we assumed that there were 6 free parameters and the others were fixed based on literature estimates. And that's a very common situation to have, right, is that you're developing a model and some of the parameters in your system you can approximate pretty well based on literature estimates, but then you have some that are still free. And the main part of this model that we're trying to constrain for this test case is the modified Hill function that basically describes the transcription rate of the reporter as a function of the transcription factor and the inhibition from the DNA binding domain. So hopefully these type of equations look very familiar to you. And basically we produce our data from this model. We forget that we have the model. And then we say, okay, we're going to develop the model to describe this data and this system. So this is what's going on in the background. And this is also going to be what I mean by the model that we're trying to fit, specifically the parameters in this portion. Okay, so here's again, the overall development process. So we start by formulating our model. And the two things that I actually think are very useful about this framework are steps 1 and 3, because normally I think we think of perhaps model fitting and evaluation as the formulate the model, fit the parameters, and then perhaps compare between candidate models or assess whether the model fits the data well. And I think these extra steps 1 and 3 add a lot of robustness to the workflow if it's not something you were doing already. Okay, so before we can begin evaluating the model, we need to figure out why we're modeling in the first place. So the first thing you need to do is write down what your modeling goal is. So in this case, we have a pretend modeling goal of being able to describe the effect of each of our inputs. So in this case, that's the DNA binding domain, the transcriptional activation domain, and the ligand, describe the effect of each of those on the reporter protein, which is the readout. And we generate our pretend data or our training data, which is a dose profile of the ligand dose or the reporter expression as a function of the ligand dose. Then we write down our model, and in this case, it's the same as the reference model that actually produced the data, but we're pretending that we don't know that that model is the correct thing. Okay, so in order to fit the model, we need to have a cost function, and so we're just using sort of a standard cost function. So the L2 error between the experimental data and the model as a function of the parameters, and we want to minimize this cost function as a function of the parameters. In this case, we have a normalization by the measurement error. And so the idea is that if we have a model that well fits our data, we can use it to achieve our modeling goal of describing the effect of each input on the readout. So now our goal is to fit this data to a test model. And the challenge is that without really knowing a lot about the system, this cost function could be very simple and have one local or one global minima, or it could be increasingly complex. And especially in biological systems, it could have whole regions in parameter space that are very flat. So, at least the thing I worry about when I'm developing a new model for a new system is if I do a parameter fit, is that parameter fit, first of all, is the method I'm using to optimize to find that parameter estimation, is it actually doing a good job of navigating these potentially complex or potentially very flat cost functions? And then, of course, not just is it navigating those cost functions, but is it finding a parameter set that actually represents reality based on what our modeling goal is. So because of this challenge that the cost function landscape could be very complicated or have many redundant solutions, and you don't know this ahead of time necessarily, we need a method to evaluate our parameter estimation. And here's the parameter estimation that we are using, or that Kate's using in this work. So it's a multi-start optimization algorithm. She does a Latin hypercube global search, which has some n search number of parameter sets. She filters those to find ones with relatively low cost functions, and then uses those as initial guesses into a Levenberg-Marquardt optimization to get some number of optimized, the initialized number of optimized parameter sets. And in this particular parameter, there's many different parameter estimation methods one could use. In this particular parameter estimation method, the number of initial search seeds that you use and the number of initialized guesses from those that have a nice low value in the cost function are the two hyperparameters. And you could imagine that depending on what the cost function landscape look like, you may need to tune those hyperparameters to be effective at really exploring the space. So the big question is, how do we know that this particular algorithm and the hyperparameters that we're using for this problem are working for our particular model, or would work for our particular model? And so this brings us to the first step, post-model formulation in this workflow, which is to evaluate the parameter estimation method itself. And this is really kind of a method and model agnostic idea, which is basically that we're going to test whether the parameter estimation method is going to be appropriate for fitting parameters for the type of model that we have. So what we do is we generate a set of parameter estimation method evaluation data, and we do this by generating multiple data sets that are as similar to our actual data as possible, but from the model. So we vary the parameters in the model, and we produce the readout, the same readout that we have in our training data. And then we use just that data that was produced by the model to see, can our parameter estimation method actually find a reasonable parameter fit for that data and our model. So again, we're not yet at all using our real experimental data. We're just seeing, for this type of model structure and the type of data that we have, can we get a reasonable parameter estimate? And so this allows you to play around or tune your hyperparameters and your method to make sure that you're actually finding minima on your landscape. And there's, of course, you need to have some sort of statistical test to say whether you're saying it's working or not, and all the details of that are in our paper. But if you can't get a good fit to data that's generated by a model across many different parameter sets, you really probably need to try a different parameter estimation method or or tune your hyperparameters. And we also in the supplement try to give some ideas of places you might want to look depending on the problems you're having. Okay. So once you've figured, or once you've checked that your parameter estimation method is actually going to work for a model of your model type and structure and the type of data that you are collecting, you go ahead and do your parameter fit. There's not much to this. It's sort of what, if you've done parameter estimation, what you would usually expect. You want to check that when you do your parameter estimation, the result you get gives you a good fit to your training data. And then that also it not only gives you a good fit to your training data, but that for those parameters, all of the states that you're not measuring in your system still have reasonable values, right? You don't want some intermediate state variable going off to infinity or blowing up to something much larger than is physically reasonable. So you can kind of do a set of checks as is everything in a reasonable physical range, or the parameter estimates that you get within a reasonable physical range. Okay, so now you have a trained model from step 2. And so the next question is to assess whether those parameters are actually unique and well constrained. And just to illustrate the challenge here, you could get potentially a good fit to your training data, but because we did this multi-start optimization, what you will often find is that the parameters that are found from that multi-start optimization are actually not well constrained. And these are 6 different parameters in our system. Most of them, again, are describing that Hill function for the transcription factor. And then this is a log value of those parameters. And so you see that some of the parameters, for example, this E, which is a transcription rate, is nice and well constrained, but the other ones, even these guys, this KM is not super well constrained within an order of magnitude. This N is a little bit questionable, and especially this N is actually the Hill coefficient. So it looks like it's within, maybe looking at that on a non-log scale, that would be super questionable as to whether that's a well-constrained fit. So parameters are not unique, and this is a consequence, again, of the fact that your cost function landscape may be complex and may have multiple minima or a whole range of minima that all have the same cost function value. So another way of putting this is that changes in free parameters may not affect the model fit to the training data, but you could care about it, right? It could affect predictions of other factors in the system. So how can we check whether this is going on? Well, a really nice method, and again, we did not come up with this method. It really, the what Kate has done is put this together into a really nice pipeline, is something called parameter profile likelihood. And the idea is basically that you take each of your parameters, which are these theta I, you systematically vary that parameter, decreasing it and increasing it, and you redo the parameter estimation with that particular parameter held at those particular values. So start with the parameter, take a step in the parameter in one direction, refit, and then keep stepping until either you reach some, you've gone far enough that you don't think it's physically reasonable, or you reach some threshold in your fit. Excuse me. So what this does is, if that parameter is well constrained, as you vary it, no other parameter will be able to compensate, and so you'll get an increase in your cost function in a well-defined minima. So that is what we call identifiable. However, if there are other parameters in the system that you're refitting every time you move this parameter, it may be that those parameters can compensate. It may also be that the noise in your data is too large. And in that case, you may be in what is called a practically unidentifiable regime, where you don't have a well-defined minima and you never cross some threshold of your whatever statistical test we're using chi-squared is. And then there's another thing that can happen, which is just if the cost function is completely flat as you vary that parameter, that indicates that there are some other parameters that are totally able to compensate for that parameter that you're varying. And so you can just keep varying that parameter as much as you want. The other one gets refit. And so you have a whole set of solutions between at least two parameters that all satisfy the same cost function value. Okay, so this is the thing I like about this method is that it is more informative than a local sensitivity test, but it is not as costly as trying to do some sort of totally global sensitivity test. So it's getting you closer to a global sensitivity analysis without really doing a global sensitivity analysis. So what does this look like for our test case? So the first time Kate fit our model to our pretend data, actually only one of the parameters, this transcription rate, was identifiable. All of the rest of them were either totally structurally unidentifiable or practically unidentifiable. And so the first thing that she did was she said, okay, well, what if we measure two more states of the time series of two more states of the system? So now we have more data to fit our model to. And I don't remember exactly what so before she was measuring the ligand or the dose response to the ligand concentration. I think she then started measuring the dose response to one of the other components as well. I don't remember which, but when she did that, she was able to uniquely identify two more of the parameters. So now there were only 3 parameters in our 6 free parameter system that weren't identifiable. So this is the same plot. So next she focused in on this M and B parameter. And I'm not showing you the details, but basically she found that when she varied these 2 parameters, they compensated for each other. So there was a linear dependence between the 2 of them. And so she replaced, by looking and examining the equations, she did a model reduction step and replaced M with M star, which is actually M over B. And that relates very well to a standard model reduction for Michaelis-Menten type kinetics. So now she has only 1 free parameter left. She did essentially another model reduction, which is a little less sophisticated than her replacing M with M over B. It was basically because this K bind parameter doesn't have any effect on any of the states that we're interested in the system. She just set it to 1. So now it's no longer free, it's just set to one value because it doesn't seem to have any effect. And so now she had a totally constrained model. So once she had a totally constrained model, there was a final, well, she actually had a set of models, right, from each of those different steps. She went back and compared the models, and you can use many different methods for this: cross-validation, Akiyike information criteria. But the last thing you're going to want to do is compare those models and evaluate again if it is actually giving you the information that you started this whole model development process for. And as I said before, if at any of these steps you're not getting a good fit, or you're not getting what you want, you may have to go back, reformulate the model, or reevaluate, or collect new data in order to make things more constrained. Okay. So I really like this framework. For me, it doesn't address, and I think it's very useful and very powerful, and I especially like the parameter profile likelihood and double checking that your parameter estimation method is robust. But it doesn't address one of the elephants in the room, which is how do you formulate the model to begin with? And this has always been part of the art of modeling, of knowing what is important to include in your model and what to leave out. And so most of the rest of this talk is going to be about how to do that in an automated fashion. And the main reason we'd like to do this in an automated fashion is because building a combinatorial set of all possible models that have all possible types of interactions in them, it would just be too expensive to test all of those models, even if you weren't doing something as rigorous as the game's framework. So for example, if we had some system that was governed by mass action kinetics, and we wanted to write down all possible models for the connectivity of the network and all of the dynamical interactions that could have on that network, if we enumerated all of those models for, say, 3 different populations of species out to second-order polynomials, that would be 10 to the 9th models that are combinatorial sets of each of the terms possible to include in that model. And that's too many things. And I'm being a little bit over the top in that, usually for most systems, you wouldn't necessarily include, there would be some screening out you could do based on your knowledge of the system. But what I'd really like is a method where we have to do as little screening out as possible. So, or maybe another way to put it is, we can really evaluate more possibilities in our model structure in a more rapid manner. Okay, so how can we do this? Well, this gets us to this idea of sparse model selection. So how do we down select terms within these models? And I should say there's a large number of different methods that are used for model inference or construction across statistics, machine learning, and dynamical systems. And I'm going to tell you about just one framework. Specifically within biology, I would highly recommend this Marbach paper from 2012 if you're interested in methods for inferring connections within biological networks. I think it did a really good job of comparing. I mean, this is a 10-year-old paper at this point, but it did a really good job of comparing and benchmarking different regression-based, Bayesian-based, mutual information and correlation-based, and other type of models. And so if you're trying to get entry into the field, this would be a really nice paper. One of the things I thought was very nice was that they looked at how well these methods worked, depending on what the structure of the motifs in the network were. So thinking about linear versus nonlinear functions, thinking about binary inference versus functional inference, and maybe one across the board thing that always makes things better is if you have perturbations in your data. So you always get better inference if you have perturbations or more information in your data. Okay, so the method that I'm have been working with and is a nice structure for thinking about this sparse model inference is a method called SINDY, sparse identification of nonlinear dynamics. So this is a method specifically if you have time series data of different states in some model and you'd like to build a dynamical systems model to explain the connections between those species. So here I have time series data from what are nominally based on the diagram I've drawn to factors in some genetic network. So the first thing you need to do is build a nonlinear library of candidate functions. So this is basically saying the derivative of each of my state variables is equal to the sum of some unknown coefficient times a nonlinear function of all of whichever of my state variables you like. So to make it a bit more concrete, this could be mass action kinetics. So monomials in x1 and x2. So x1, x2, x1 squared, x1 times x2, x2 squared, and so on. And the important thing here is that there are no unknown coefficients or no unknown parameters within these nonlinear functions. So all of the unknown parameters are out front. The system is linear in those unknown parameters. And as long as you have that, then you can write down this set of equations. You can plug in your time series data into these functional forms, and you just end up with a linear algebra problem where you'd like to estimate what these coefficients, these unknown parameters are on your different functions to reproduce the dynamics that you've observed. So if we just did parameter estimation on this system using, for example, least squares, most of the terms in this library would appear, the parameters would be non-zero, even the ones that shouldn't actually be there just because of overfitting to noise. So the last thing we want to do is we want to impose our idea or our knowledge that most of the parameters in this system should actually be zero, meaning that most of the terms in this library should not be there. And a way to do this that is very simple to understand, that computer scientists don't super like because it has weird convergence properties, is to just alternate between a least squares parameter estimation and hard thresholding. So basically you fit, you use linear algebra to fit the parameters in this system. You take all of the parameters that are below some threshold value, you set them to hard zero and throw them out of the library, and you refit with the remaining terms. You do that iteratively, and it tends, especially for dynamical systems, to converge very quickly to a solution that is, if you've got pretty good data, the right local minimum. So, and you notice I threw a little bit of caveat in there, the right data, so on. It's not super straightforward when this will always work. But the idea is that, okay, you have now a free parameter, right? We don't know ahead of time how many of these terms should be in the system. So if we vary that threshold or vary that parameter, when the threshold is very small, you're going, or let's see, when your threshold is very small, you're going to keep most of your terms. And so then there's going to be a larger number of terms in your model. And as you increase that threshold, you're going to throw out more and more terms. And at least in practice, hopefully the correct model is somewhere in there. To figure out what the correct model is, you need to do, again, some sort of cross-validation or information criteria-based approach. But in the end, you get either a single model or a set of candidate models with a subset of the terms from your library. Okay, so this sparse identification of nonlinear dynamics method was developed by Steve Brunton, Josh Proctor, and Nathan Kutz at University of Washington. And I got to work with them as a postdoc right after this paper came out. So I worked on a couple different extensions to this method. So one was actually using information theory to do the validation of the models and show that you could then, from the set of models of varying sparsity, select the ground truth model for a variety of different cases. I also worked on a method that was, so the previous method, the original Cindy that I just showed, doesn't work if you have rational functions. It only works if all your terms are linearly added together. So I worked on a method that allows us to do mass action kinetics with rational functions. So that's much more appropriate for metabolic networks and regulatory networks. This method has now been, is now a little outdated actually. So I'd refer you actually back to some of Steve Brunton's work where they showed that the particular optimization I was using, while very cute, is not as fast as framing it in a slightly different way. And then a problem that I'm really interested in, we had a paper that was on hybrid systems. So if the model structure changes during the measurement, can you detect when those changes occur and still do model selection? One of the big problems with all of these methods, as I was sort of starting my group at Northwestern and thinking about how to apply them to biological systems, is that often in these systems, we can't measure everything. And all of the methods that I just talked about, Cindy, and all of the various extensions I talked about, require that you measure all of the important state variables that are dynamically coupled together. So what my group has been working on for the last 4 years is a method for handling systems when there are hidden state variables. And how, one question is, okay, if you aren't measuring everything, why on earth should you be able to recover dynamics of something that you don't measure? And so I'm hoping everybody in the audience hasn't seen this before, because it's a super awesome little bit of math that I like sharing with people. So in 1981, Dawkins showed that if you have a system, and specifically he was looking at chaotic systems, so this is the Lorentz system. If you're not familiar with it, it has this nice butterfly attractor. So if you start somewhere near this attractor, it will go to the attractor. That's why it's called an attractor. And then it will wander around on the attractor. And it has the main components of chaos, which is that you can't predict the time series of the behavior on this attractor, even though there is this structure in the system. So here are the equations. And here is a time series of one of the state variables, x, as a function of t. And so again, you can see this looks very kind of oscillatory and noisy, and it's really hard to tell from a single state variable time series that this thing has the structure that it has when you look at it in 3D. So what Dawkins showed was that if you took just one of the state variables and you plot it as a function of t, as a function of t minus some time delay, so maybe 1 or 2 or 3 steps back in the time series, and a function of 2 times that time delay. So let's say we took our time delay as 2 steps back in the measurements, that would be 6 steps back in the measurements. So if you plot it in these time delay coordinates, you can actually recapitulate the structure of the attractor that you would have gotten from observing all 3 state variables. Okay, so what this says is that all of the structure of our equations y and z are actually contained in the time history of x. And this is because this is a highly coupled system. This also consequently is a reason that Granger causality fails for these systems if there's no injected noise. Because they're so highly coupled together, all of the history of each state variable is contained in every other state variable. There's a little bit of a symmetry problem with Z, but I'm going to ignore that for now. Okay, so this is really cool. Basically what Helena, Sasha, and I got interested in was Sasha had worked for her PhD on a method to do parameter estimation for these type of chaotic systems with hidden variables. And so we thought, okay, well, let's try using this awesome parameter estimation method that works for hidden variables to see if we can also do model selection with it. So take the parameter estimation method and impose the sparsity constraint that most of our parameters should be 0. So here's our setup. We have some, again, some coupled dynamical system that, has some unknown function f. But in this case, there's 3 state variables. F is moving each state forward in time, but we only, through our measurement function h, measure 2 of them. So you can see in this case, we're measuring, I think we measure x and z and don't measure y. And confusingly, we're then going to call those things that we do measure, measurements y, with our hidden variables x. So now we're going to formulate a library to describe this dynamical system. And our library is going to be, we are going to assume that we know the dimension of the system. There are ways to figure out what the dimension of the system is using, again, a time delay embedding type thing. And we build a library of sets of function. In this case, because we know our Lorentz system lives in polynomial space, we're using polynomials again. So we don't know which of these terms should be selected or what the parameters are, and that's our goal. And another way of putting this is that we want to minimize this log likelihood function. I will now break this log likelihood function down for you. So this log likelihood function has a measurement error term. So this is comparing our measured data to our state variables predicted by our model, but only the ones that are measured, right? So in this case, I said we measure X and Z, but not Y, transformed through our measurement function, which we assume is actually just additive Gaussian noise. And so H would just be, it's just X. So we have our measurement error, and then we have our model error, which is taking the difference between the model at the next time and the values at time t+1 and comparing that against the prediction of the model at the using the previous time point. So this model error term is actually the part that does the coupling between the different state variables, and therefore has this idea of Tauken's embedding theorem, that the information in our unmeasured variables is actually contained in our measured variables, but we're doing it just by enforcing the model. And then the last term is a sparsity term, which is just saying that the L1 norm of our parameters, so the sum of the absolute values of our parameters added together, weighted by some lambda, we want to minimize that. Okay, so this is a difficult cost function to find the minimum of. I'm illustrating this by showing you, we simulated some data from the Lorentz system, and then we did parameter, like we essentially evaluated this cost function, assuming there were two extra terms that should be zero hanging out. So this is a function, a plot of the cost function as a function of these two extra parameters. And basically, the reason this is so hard is, imagine trying to find the true global minima in this system, which should be at P21, P11 equals 0. This is a very hard cost function to navigate. So how do we do this? So this is a method called variational annealing, and we did not come up with this method. Again, a group, here's the reference for the original variational annealing method that did not have the sparsity term. So how this works is you start out by having the model error term set to 0. So you essentially say that RF is 0. Then you have a nice convex measurement error. You slowly turn RF on, and now your cost function gets more and more bumpy. But because you started near 0 in your measurement error, you can stay near that global minima, keep enforcing your model. So dial that up and up and up until everything converges. And then hopefully, you want to, again, multi-start this, but hopefully one of the minima you sample is the global minima that was relatively close to your measurement error minima, but with your model fully enforced. So then we add, and I should say this parameter estimation method has been used for training neural spiking models. So I do think the method that I'm talking about right now could be directly applied to some of those neural spike models if somebody was interested in doing parameter estimate or model selection for those type of systems. Okay, so then what is this sparsity term doing? Well, what it's doing is you essentially do the same thing as we did in Cindy. You increase it, and it forces more and more of the parameters in your system to go to 0. And so this minima ends up, in multi-dimensional space with more and more of your parameters forced to 0. So we essentially need to iteratively do our variational annealing, where we enforce the model, and our sparsity constraint, where we set more and more of our parameters to 0. I'll refer you to the paper for exactly how we put that algorithm together. We tried a bunch of different ways, and the one that we published is the one that worked, but there was a little bit of art to getting it to work. But as we vary our lambda, we have sets of models which represent the multiple minima, and you can see a general trend that the number of active terms in these models decreases as we increase the penalty on complexity. Okay, so we have not gotten this to work for biological data, and I'll tell you why in a second. But the test case that we used was this circuit data, which the Blakely et al. Originally designed the circuit to produce something that looked a lot like the Lorentz system. So we thought this was a good test case, but to actually use experimental data rather than purely synthetic data. So we took our data, we constructed our library, we plugged it in to our algorithm, which we're calling DAISY, and we found a set of models. And I'm going to at least partially declare victory because one of the models that we found had the same structure or a very similar structure to the Lorentz system. That's victory asterisk because the model, there are also a set of models which actually have stronger statistical support than the Lorentz structured model, which say there should be a couple additional terms. So unfortunately, we don't have experimental access to this circuit to test whether those terms are real, but there's, at least my intuition is that it's pretty easy to end up with some of those extra terms due to parasitic resistances and things like that. So in many ways, our model selection method is succeeding in that it is suggesting additional terms that should go be tested experimentally. Okay, so when we took, and I should say, the big victory in this whole process for me is that Tawkins, I don't remember now when that citation was, but you know, in 82, he said we could recover the Lorentz system. We should be able to reconstruct the Lorentz system just from with a hidden variable. And now in 2022, we've been the first group to actually rediscover the equations for the Lorentz system with a hidden variable. We would have loved to do it with only one state variable instead of two, but we did some calculations to figure out how much more, how many more seeds we would need. And basically, I don't think we can do that without some optimal manifold sampling, which we don't have a good method for doing. So basically, we need to do better experimental design, or else the cost function it has just too many local minima to navigate in a cost-effective fashion with only one hidden variable. Okay, so back to biology. So we had this method working, and I was like, this terrible Lorentz chaotic system, which there are many systems, I think, in biology that, you know, especially like thinking about modeling the heart, thinking about modeling in neuroscience that have chaotic behavior. It happens that I'm not working on those type of systems, but I thought, okay, this thing is absolutely a terrible cost function landscape. If I apply this method to my metabolic systems, this method should have an easy time finding solutions to this system. And it turns out, no, that's not the case. And this, you know, this is what happens when you are not formally trained and discover these things on your own. But basically what I now understand is that metabolic systems have the problem that many ODE type systems have, which is that they often have identifiability issues, as I discussed in the previous section of the talk. So there are multiple parameter sets so that will produce the same cost function. And the type of optimizer that we used in our method is actually really, really bad at navigating flat landscapes. It's much better at navigating these type of very convex, bumpy landscapes. The second thing is that chaotic systems, because they will wander all over the manifold, they do your sampling for you. You just have to run time longer and you get more and more sampling of your system. In metabolic systems, you run your time, they go to you run your system, they go to a fixed point and they're done. You can't just take more and more time samples and understand the dynamics better, you actually have to design different experiments with different initial concentrations in order to get a good sampling of the dynamics of your system. So basically, my group is now working on methods for both pieces of solving this problem. One is reselecting what the optimal optimization methods for systems with flat type cost functions are going to be. And then second of all, doing experimental design to make sure that we have the right data to actually constrain those systems. So that gets me back to my overall picture. And I want to thank my group, especially Helena, who did most of the work on the second half of the talk, and also my collaborators, who I don't have their pictures, but Kate for doing all of the work to get games up and running. And thank you very much for listening.
Okay, let's congratulate Professor Newman. Thank you, Neil, for the very nice talk, very nice presentation of your work, of your group, and all the thoughts that have been developing since you was a postdoc and then a professor in Northwestern. Do we have any questions from the audience?
And while people are thinking, again, I'm very happy if students or postdocs want to get together and email me to schedule a time to make it a little bit more like if I was there in person to actually discuss.
Yeah. Okay. Yeah. No, if...
But I don't want to stop people from asking questions now. I'm of course happy as well.
Okay. Just a curiosity. Games is programmed in Python, I guess.
Yes.
It's a Python API like this,
yes.
Or do you have some windows and interface,
No,
a user interface?
It doesn't have doesn't have a user like a nice GUI interface or anything like that.
Yeah.
It's definitely meant for somebody who has some comfort with programming. But the idea is to make the workflow very systematic, and I think putting together. But yes, the barrier to entry is not zero. You have to have some familiarity with how to do ODEs.
Programming 101, numerical methods 101.
Yeah, and I will say Kate just released a new version, which I think is working well with the newest version of Python, and it's like being actively updated. So if anybody has trouble with it, please actually email her, and she'd be happy to help.
Okay, what was not clear for me, I guess I did not understand the connection with the part that you said at the end of the first part. You said, okay, but now you must have a model to start with. So right. and the connections with the last part of the talk, you plug in your methods in the beginning of the games pipeline. Is this?
Let's go. Yeah, let me go back to, because there's kind of two places that model selection could come in. So the first would be in formulating the model, right? That maybe you have a large set of possible terms that could be in your model, and you would like to down select those terms to have to formulate a model as your initial model to try out, right? But the second
model or set of models. No?
Yeah, you're right. It could be set of models. Okay. Yeah, it could definitely be set of models. And then the second, yeah, you're absolutely right. This would be, I think of it as first you do the model selection, which is just selecting the terms, and then you still should do some parameter estimation after that to fit each of those model structures. The other place, though, that this, depending on how many candidates you have, model selection could be a way of doing model reduction. It's not the only way of doing model reduction, but it could be a way, depending on the complexity of the model that you formulate at the beginning. If you find you have many identifiability issues, you could at least in principle imagine using sparsity as a way to do the model reduction at the end during the kind of in between these two steps of assess and compare.
Okay, there's also this your talk just remembered me another question. But what do you mean by model reduction? Is something like POD or other data-driven surrogate models like Gaussian process or something like that? What do you mean by
so in this case, I actually mean model reduction in the sense of looking at our ODE's structure and making decisions about these ODEs. So for example, if you have a case where this inhibition is negligible, right? That's not a case that comes up in the system, but you may imagine removing this term from the model if the inhibition is always negligible. That would be a form of model reduction. So just removing a term. Another form of model reduction, which Cindy can't help with, right, is setting certain things to pseudo-steady state. So saying one of these sets of equations is in equilibrium or in pseudo-steady state, and then replugging the solution to that back into the rest of the system.
Okay. Do you have some algebraic tools also to guide this? Oh.
Yeah, we don't currently, I mean, this is something that I think we need to build out, is we don't currently have automated methods for doing, especially the pseudo-steady state. There's another group of methods from Sethna's group. It originated from Sethna's group at Cornell, and he has a couple students, Transcription and Makta. I'm not sure which ones were students and postdocs, but there's a couple of people who've proliferated out, which work on automated methods for detecting when you have, when you can do a model reduction, but they still have to do it manually. So they haven't quite gotten the step, like none of us have quite gotten the step of doing the algebraic manipulations in a manual or in an automated way.
Okay.
And I'm happy to point you to the references if you're interested.
Okay. Maybe I write to you. Okay. Let's congratulate. I'm sorry. Is there any question? I guess I took all the time. Okay. So as Professor Neo said, she is available for the students to, if you want to contact her for a schedule for a later chat for some work. Okay.
Thank you very much.
Thank you very much, Neo. So we have a half an hour stop, and then we resume with Professor Kariokana. Okay, I guess I pronounced it right, Kariokana. Okay, so see you in half an hour.
So we are now back. Care. Great. Let's see if you can.
Yes. Okay, I'm going to.
Yeah, let's see if you can share your screen, please.
Yeah. I lost the connections. Okay.
Okay, so
Yeah. It's okay, Professor?
yeah, but I think that this is not in full screen yet. Is it?
No? Yes, is it?
Okay, great. So
Yes.
it's my pleasure to introduce Dr. Karen Del Carme. She's a technologist researcher at the National Scientific Computer Laboratory of the Ministry of Science, Technology, Innovation, and Communication, young scientist of Rio de Janeiro State. She has a postdoctorate from the Systems and the Computer Engineering program at the Alberto Luiz Coimbra Institute, UFRJ, a doctor and master degrees in cellular and molecular biology by the post-graduation in cellular and molecular biology of the Institute Oswaldo Cruz Fiocruz. She has a title of chemical pharmaceutical and bachelor in pharmacy and biochemistry from the Faculty of Pharmacy and Biochemistry of the Universidade Nacional Mayor de São Marcos. She has a title of pharmacist. Currently, her main area of expertise is bioinformatics and computational biology, working mainly on the following topics: comparative, evolutionary, and functional genomics, phylogenomics and phylodynamics, development of mathematical methods and computational tools applied to genomics, and the large-scale scientific data analysis in high-performance processing environments. So, Dr. Karen, thank you again for accepting our invitation. The stage is yours.
Okay. Thank you so much for this opportunity, Professor. And then I also am going to, okay, I'm going to fix, okay. Thank you so much for this opportunity, Professor. And then it's my pleasure to be here for the first time in this opportunity. As the presentation of the professor, I'm Carol Caña. In this opportunity, I'm going to present this modeling efficient phylogenetic and RNA-seq scientific workflows and my experiences in the Brazilian petaflopic supercomputing system, and in this opportunity on the Santos Dumont. well, this is a multidisciplinary research that involves different researchers at LNCC and other different collaborators, Brazilian collaborators and international collaborators. These are there are not space for more photos, but there are there are the collaborator and students that are more involved in this result that I'm going to present in this opportunity. This involved, and thank you so much to my lab, Laboratory of Bioinformatics, to CINAPAT, CENEPAT, the Santos Dumont, and all these universities and research institute. Everything about me, the professor introduced, but I need to reinforce that the interdisciplinary involvement that I have. I studied biochemistry in Peru and here in Federal University of Rio de Janeiro, and after that I involved with my informatics research at Pio Cruz in my post-graduation. And when I started in the computational science department with Marta Matoso at COPY, I started to develop some scientific workflows. Then I can perceive the importance of this type of point of view, how my experiment, in this case bioinformatic experiments, but any other type of experiment can be used in that, can be use these technologies of scientific workflows, of scientific workflow management system, of database management system, and I remember that I started with clouds and using clouds for before the introduce of the supercomputer Serum in Rio de Janeiro, and then it was an opportunity to explore Amazon or Azure, and then this is the first HPC environment that I was involved in developing the bioinformatic workflows. And after that, when I entered as a researcher in the LNCC here in Brazil, in the same year we host the Santos Dumont and the Santos Dumont at the LNCC and the Lobo Carneiro at UFRJ. And then we have this importance to stress the machine to better understand how about the behavior of the comportamental experiments that are executed in this platform. And for that, I am going to explain that, because I'm going to focus more than in this scenario. This my involvement with the biological scientists and specialists. I am going to. It's very, very important to develop this type of case of study, this kinds of problems that need to be resolved. And then after that, I'm going to focus this presentation to involve my bioinformatic expertise with the people that work with the Santos Dumont, specifically how I use this environment, this computational environment, how I can introduce all my experiment, how I can, for example, deploy the bioinformatics libraries or transference of any data, etc. And then, my history starting with bioinformatics, and after that with designing the bioinformatics workflows using point of view of the scientific workflows, and after that how I use in the first case clouds that is a very nice case of study to develop some phylogenomics workflows, and after that how I can move my experiment to Santos Dumont, and I explore more about the performance, the scalability of these analysis that were performed based in bioinformatic workflows, and how we started to develop the gateways, that is a bioinformatic, like a portal that contain different type of bioinformatic applications and workflows. And then in the first times we have one class of study, one problem, for example, phylogenomic workflows. And then after we started with, okay, we need to deploy that in Santos Dumont, for example, who are the bottlenecks that are presented in the executions, or how is the behavior when I execute that about, for example, the performance or the scalability, and using different architectures in Santos Dumont. And when we optimize the workflow, this workflow is automatic, and with this workflow, we will explore more this workflow, and we deploy that in these gateways that is open for the bioinformatics community, and it hosted in the LNCC. And wow, this I'm going to present like my title, two types of case of studies. The first one is the phylogenomics, and the second one is more recently the RNA-seq. And in phylogenomics, I started with that from my PhD, and then this is my one of my favorite analysis. And then well, we started with the analysis of phylogenomics as with studying the protozoan genomes. What about the evolutionary history and the relationships of the different genomes of the protozoans? And then, but 10 years ago was okay. It was difficult because the genomes. More of the genomes of the protozoa that are considered neglected tropical disease were not finished, or they are not available, and we have a little problem to the reconstruction. But using different type of phylogenomics techniques, we started to scripting the code, and this code. Okay, we're thinking about that. The first step that I need to. In my algorithm, for example, is the construction of the multiple sequence alignment. The second one is for example, the selection of the best evolutionary model, and the other is which one of the algorithms and software that I'm going to use and is going to be introduced in my script. And these decisions were very, very important, and they're more focused about, okay, which one is my case of a study on phylogenomics and on protozoa and on the utilization of protozoan genomes. But obviously this is before my experience with the computational science group, and then it was interesting because I remember my script in Perl in Python, or I think it was in Python, and it's automatic, and it ran very well, and it was very, very, very nice. But in this case, for example, we reconstruct the evolutionary trees of phylogenomics. And in red, for example, there were included the genomes that are not complete. And in the second one, in the blue, for example, there are some genomes that were introduced in my experiments with complete genomes. And with that, okay, I introduced two types of problems. Maybe yes, I am comparing, or I want to reconstruct an evolutionary tree, but one complete genomes and one incomplete genomes. But with that, the evolutionary history, it was very, very similar with the gene tree of the protozoans. And then this study was the first phylogenomic experience using that, but not starting yet with the workflows definitions. And then, okay, I'm going to in this case, I'm going to present this panoramic in the pandemic in these two last years, and then but I'm going to. Well, yeah.
I'm sorry to interrupt. I think your slides are not advancing.
Not?
We're only seeing the title slide.
Oh, for example, I am in the phylogenomic analysis. Not?
Yeah, I'm sorry, we're not seeing that.
Okay, I'm going to share again. Sorry about that. Is advanced now?
Yes, now it's working. Thank you.
Okay.
Earlier.
Thank you so much for that. Okay, okay. But I was starting with the presentation of my cat. Okay, thank you. And well, in this this is a group of different type of papers that are related with the starting phylogenomics and genomics studies related with the COVID. But in here, I'm going to put this presentation more in methodology and more in bioinformatics, because I am going to link that with the use of the HPC environment and technologies. And then we can say in different type of colors that, for example, in here, this is the phylogenomic reconstruction using SARS-CoV. It's related with which one of the data, about the data, and when it's starting, it was starting in 2020, for example. Okay, there was not complete genomes and starting this type of study. But in the studies in general, the state of the art of the phylogenomics software were used. For example, there are more publications using Ruxmel and Exomal and BEAST, EK3, and there are some state of the art that is very, very interesting, because there are based on maximum likelihood and Bayesian inference, but also Ruxman, for example, it was the code is changing, also is evolution, is on evolution, because no Ruxman is adapted for supercomputers. And then I'm going to explain that in my posterior slides. And also in here, for example, Mr. Bayes or BEAST also have a library that in joining this code with the library is supported to the execution in supercomputers on the HPC environment. Okay, and on the other hand, there are not only the phylogenomics or not only methodologies is important here. Also, it's very interesting that other type of metadata was introduced here. If we are going to reconstruct the phylogenomic analysis of the evolutionary tree of one organism, for example, as I presented as protozoans, for example, if we included more than genomics data, for example, localization, time, etc., we include more information, and this for phylogeny and that phylogenomic workflows or analysis are in way to, for example, phylodynamics. That is what's very, very interesting, because for that, when we use this different metadata, we can reconstruct the distribution of this type of virus over the world, for example. And but as I told you, I'm going to analyze more about the methodology, which one of the software, the algorithms, and the infrastructures that I'm going to use, and how I'm going to integrate that. Well, the first one is like the database for COVID, for example, as in other type of genomes. For example, here in Brazil, we have a very dramatic problem about the epidemic, pandemic, and from the neglected tropical disease. Not only, for example, today the COVID is more controlled, but we have problems with trypanosomiasis, with leishmaniasis, with other type of neglected tropical disease that are endemic here in Brazil. But in here, for example, GISAID was a specific database that centralized all the information about COVID, and it was very, very different. It very interesting, because in this you can download the information, but also you can download not only genomic information, but about metadata, about the distribution. You can visualize here in one centralized web, or in this case, web portal, for example. It was very, very nice. Here then there are more a little examples about the phylogenomics and phylodynamics. For example, if you use some metadata as localization or time, you can deploy that in one software for visualize about the distribution and the reconstruction for this. In this case, it was dengue in Brazil. And then in here, for example, which one is about, what about one type of paper, of very interesting paper about COVID? In here, there are points that we need to observe. The first one is which one, what about the data, which is the data for the bioinformaticians create the data to use these genomes of sarcophagus, for example, to introduce that in the workflow for phylogenomics. The first one, well, okay, I'm going to download my data from the GSAID, for example, this type of data. There are in phylogenomics, there are some rules, for example, the orthology and etc. If we cover all these features, okay, which one is the methodology that I'm going to use? In general, it's used the maximum likelihood. And in this case, the maximum likelihood is more represented from this type of software, the Ruxmel and EK3, that both of them are have versions with that can be executed with threads, with MPI, or also in the hybrid, using threads, both of them, threads and MPI. And this was okay, because we can benefit from the parallelization and the distribution of my jobs, especially in this type of HPC environment. I'm going to introduce a little more some concept about the algorithms. Well, the first, when we model one problem in phylogenomics, we started to recognize every step and every data that is related with this step. And if I for example, I have the differences, the set of steps. Okay, the first one, I'm going to download the data. I need to filtering. I need to align these genomes. I need to trim in. I need to construct the phylogenetic trees and other type of posterior phylogenetic analysis, and all of them need to be very reviewed, because we need to understand about the features of the data, which one of the software that I'm going to use, the software are available, or I need to construct a script, for example. And this one, for example, in this part, I'm going to use maps for the alignment Y. In this part, I'm going to use a trimming after the alignment. Why not? I am going to use the total sequence. Okay, in the phylogenetic inference, I'm going to use model test or model generator for reconstruct the evolutionary model. And after that, I'm going to use EK3, but which one about the EK3 or the Ruxmel? And then these are the decisions that we need to fix. And when we model this part, I can model that thinking in scientific workflows. And then after that, until here, I'm talking about the experiments. Okay, my experiment have these steps, use this type of software, and etc. But the important thing for the reconstruction of the tree, for example, is I need to better understand what about my software, for example. Obviously that the code is developed for experts. In this case, Alexander Stamarakis that developed the Ruxmel, but I need to understand which is the feature that is related to the deployment of the jobs, for example. I need to better to understand about which one are the optimization in the code that allow that Ruxmel execute in supercomputers, for example. Nor than and in the law with all the information, but I need to have that information because I am going to use the parameters that are related with Ruxmel. For example, if I know that it exists an threat version, I know that I need to fix this parameter in Ruxmel to be executed in an HPC environment. Well, this is my phylogenetic experiment, my basic phylogenetic experiment. The first time is my alignment, and the second one is my model, my evolutionary model, because I'm going to execute that with maximum likelihood, and then some, for example, execution with Ruxmel, and it's okay. There are they are the command lines that I'm going to use in my workflow. About the running of phylogenetic analysis. Well, there are cases that we need to model in workflows, and after that I need to think about which one is the environment that I'm going to use. I'm going to use my desktop or my supercomputer, for example, but if I am not a supercomputer and I can use, for example, a private and public cloud, maybe I can do that. After that, these are the steps that I need to follow, and I need to think about the storage of my data. In this case, before we use Amazon, but in this case, with the opportunity to use the Santos Dumont, we can use the supercomputer. And after that, at the final of my modeling, execution, analysis, are my tree that are going to be used for reconstruct the evolutionary hypothesis about the evolutionary history of my organisms. And scientific workflows are abstractions. There are abstractions that follow that my steps that represent a chain of activities that are related with a scientific experiment. There are two types of concepts. The first one is the scientific workflow management system. For example, there's one that Swifts, a Tabaxis, a Cumulus, Galaxy, Bistrails. The one that are using in this moment is a Parcel, for example. And that also we're thinking about, okay, I'm modeling my scientific workflows, and maybe I'm going to use a scientific workflow management system to manage this workflow, and this system is going to be the connection with the architecture of my HPC environment. I'm thinking about my variation of the workflows. If I use, for example, one type of software, and in the other opportunity I'm going to introduce other activity or I'm going to shift for other software, I need to think about what about the variation and how I'm going to store this type of variation of my workflows. Then provenance is related to metadata and data. All this information is very, very important to modeling my workflows. Then when I'm thinking in modeling workflows, I am thinking about modeling my data flows. Which one is the data that entered and which one is the data that is generated for one step or activity. This is my experience with the clouds. Clouds for comparative genomics, to phylogenomics, for evolutionary hypotheses about hypotheses, and for molecular modeling or for document modeling. But the important thing here is that each one of these experiments have different variables or different point of view, for example, different problems, and we can resolve that modeling my scientific workflows. And after that is very important, think how I'm going to model my scientific workflow, and then how is the architecture that I'm going to use to deploy my executions. And in this case, we observe that clouds are bioinformatics can benefit from clouds from deploy the experiments in large scale. Well, here then we started with scientific workflow management system using here the comparison with say Cumulus and Hadoop for starting to observe the variations And we observed that well, here, for example, that, okay, after that, this is a very, very simple phylogenetic workflows that have only 4 activities. The first one, I'm download the database, for example, or my genomes. I need to construct the alignment. I need to test the evolutionary model. And after that, I need to construct the phylogenetic tree. But in here, when I model that, if I open each one of these box, I can observe my command line that I fix the end pairs, the parameters, etc., and cover all of that. I am going to put that covering these workflows. I'm going to use my system workflow management. And then, for example, for Cycumulus or for Swift or for Pegasus or anything, I need to in some cases, it's not a very simple task, because I need to understand about which one is the code I use my workflow management system, and maybe I need to introduce my command line of the, for example, Raxmel to Cycumulus, for example, or to Parcel. Here, when the thing is that when the workflow is more complex. If we use, for example, one type of scientific workflow management system, the thing is that now I use the Cycumulus, for example, or the Hadoop or an architecture like a Spark, and then but tomorrow I'm going to use Parcel, for example, and then now there is not a connection that, okay, I spend my time codifying here and making my script here, introduce all my bioinformatic information in this scientific workflow management system, and tomorrow I need to change that. And then we need to be very, very careful with these decisions. In this case, we model in the phylogenomic workflows. It's automatic. We use the Cycumulus, that is a system workflow, that is the system workflow that you use to manage these workflows. And about the scientific workflow management system is that each one of them has a very particular feature. For example, the Cycumulus that I use is more have a middleware that use and store automatically my data in a database. But for example, Galaxy is a web system, and then for example, Parcel is a language script that is very, very now I think it is the most useful for the connection between HPC environment and scientific workflow, but we need to know about the language that I'm using and that are related with the system workflow management system. Well, this is then when this is my one of the results is obviously that if I use, for example, in one thread, I am going to, or in sequential, with one thread or in sequential, with the sequential version of my program, I am going to spend more time in my execution. But also, if that means that if I introduce more cores, in this case virtual cores, my total execution time is going to decrease. In here, we compare two types of systems, and we obtained that with Cycumulus it was obtaining the best results. But the important thing here, the thing here that we need to observe is, okay, in this point, in 128, we observe that it decreased maybe 90% of my total execution time, but we need to observe also that for in clothes, I am paying as I'm consuming. If I, for example, use more cores, I'm paying for more power in the clothes, and then maybe we observed that in that point we spend more than approximately 90% of the time, and we don't need to rent more virtual machine here that we are paying for that, when we can obtain a result that it was nice and for my type of experiment. Well, this is until here. This was the experience in clothes using Amazon. From 8 years ago to now, the Amazon is in a framework. It's a wonderful framework with a lot of information and a lot of community. The community is very, very strong. But after that, I am going to present the experience in the scientific workflow in the Santos Dumont. Here's my institute. My institute is here at right, and then this is the Santos Dumont. And the Santos Dumont in 7 years ago was in the top level in the world, and now is considered the largest supercomputer in Latin American, Caribbean supported research. And this is in Brazil, the different type of the different center of processes that are distributed in Brazil, in all Brazil. And here, for example, there are some clusters that are in the different centers of processing here. Here in Rio de Janeiro, we have the supercomputer, the Santos Dumont and the Lobo Carneiro, and the Cinapat is the system that manages all these great. And for example, if the idea is that if for example Santos Dumont is full, I can maybe use the Cinapat or the Cinapat, for example, from the north of the Brazil for finish more process. And here in the Institute, we develop different type of web portals and gateways. And this gateway was developed for my group. That is a gateway that hosts a different type of application and workflow, the same workflows that we are modeling and optimizing. When we finish of optimize that, we deploy it in the beautiful portal. Here, this is a scenarios, a total scenarios how we start with one execution. For example, the first one is, okay, I'm going to choose the data that is going to be used in my experiment. After that, which one is which type of the performance execution I'm going to do. Okay, I'm going to for example, test the execution based on the size of my data, or for the parameters that are used, which one. Then there can be different type of performance test for each one. For example, in this case, it's Raxmel. And as we can, for example, do the scalability experiment of the scalability execution of this experiment, and then we can observe in which one of the nodes is the bottlenecks, or which one is the node, which one is, what about the behavior of my execution if I include more nodes. And we can do that in terms of the total execution time, or the efficiency, or the speed up. And this is a complete study that it was performed in conjunct with my collaborators that are the administrators of Santos Dumont. Well, specifically for the Raxmel here, starting, we make a comparison, a very basic comparison. Okay, we have the serial, we have the version that works with p-treats and the MIPI and the hybrid. And then when we use the same data with the same architectural and performance and technology features, we can observe here that we, from 1 core to 120 cores, we can decrease the total execution time in 98%. And then if I compare, for example, these three versions, Patrician, Piano, and Hybrid, the hybrid in the architectures for the Santos Dumont we obtained with this version the best performance and the best scalability, and that is the elected for the elected for the that is going to be included in workflows and is going to be deployed in my future executions. But the important thing here is, this is my scalability, but there are some points here. And we are studying what about this point, why we can see the decrease is total execution time, and then after that, it's like there is an equilibrium here, but increasing more nodes, there are some fluctuations and some fluctuations why, yes, and then this is the finite tuning that we are studying in all the workflows performance analysis. Okay, my second study was the hernia-seq and why. The phylogenomics is a very, very interesting analysis, but it's very complex because in each one of the steps or the activities, we are going to obtain different type of data, and we need to join this data and etc. But the more complex, there is not, for example, it's very, very difficult that one genomics or phylogenomic analysis can produce terabytes. It's probably in gigabytes, and in that is it. But what about the stress in the machine? And RNA-seq is a very nice candidate for that, because we use other type of transcriptomic data, and that we easily produce terabytes. And then it was very, very interesting because in phylogenomics, we study the CPU, the scalability, and the performance. But here we can study more the memory or the I/O, for example. And then after that, we move over for in one of my studies. We started with that. And then we started with that because in one visit in Germany, this Laura was producing data related to transcriptomics, but in the studies of cardiomyocytes. And then, but this is from the laboratory of toxicology, and then we discussed about the procedure and the workflow that she uses, and then she uses Galaxy, and the protocol was very, very nice. But Galaxy is a web portal. It's very, very strong, very, very nice, because we can model it easily, your workflow, etc. But if you want to explore more about the parallel and distribution of tasks, maybe you need to link this type of system to your local infrastructure, for example. And okay, the first one was understand about the study, and we needed to elect the different type of programs that are going to be included to compare the different workflow that were for compare that, and you can observe that. It was a very, very complex experiment, but we are more interested in the part that is computing and time intensive. This is the thing that we need to attack in this moment. I'm going to present this in the poster, but I need to show that my transcriptomic here is at the starting of that workflow. Other workflow, because I need to produce some data that is going to be used for a workflow that is more used to join databases, and is more data analytics. This is the posterior analysis, because now we're interested in workflows that are computer and time intensive. And we validate that. We download the workflow that was developed in Galaxy. This was very, very complex. Okay, we starting with that, we modeling in Galaxy because the activities were not connected. We connect all the activities. I remember that we needed to store that in the Ferdom, that is the database that they use it for the provenance and metadata storage. And then after that, we modeling the workflow. The workflow very, very similar with the phylogenomics workflows. We have an activities, we need to better understand what about the performance and the scalability of each one of the activities, but not all the activities. For example, we need to see about the bottlenecks, which one of these activities are considered the bottlenecks. And we observed that both tie and the agate sex was the very interesting activities that are more intensive. And well, this is for one with patients, but imagine a big scenario when we optimize that, and these workflows can be used in different patients. It's going to be the quantity of big data that is going to be generated is huge. And after that, okay, we model the workflows, but we can see the variability is obvious that it can be present in that. And then we needed to show which one is the software and which one is the procedure that we're going to use. And based it on the concept of the scientific workflows, the property of each activities can be, for example, invariant or variant or optional activity, or some activities that are that need to be present in my workflows, and depend on that. This is a concept of line of experiment that are used in computer in software engineering, but also can be extrapolated to bioinformatics, and we can classify the activities to do that, and we observe that all my workflows can be reduced to that, to 3 or 4 activities that are going to be used and going to be representative to modeling my workflow. And then after that, we validate also if I produce the Galaxy with the activities that are not connected, or the activities that, or using the workflow in Galaxy, or using the workflow in the parcel that we model it in nowadays with this system. We needed to validate that with the results that biologically not affect this result, this biological results. And well, I'm going to, are 2 articles that are one accepted and one submitted about our parcel RNA-seq, because this is our workflow of RNA-seq that was modeled in parcel and was executed in Santos Dumont. We used that for the single carbon-associated RNA-seq data. We use parcel, we use it at an infrastructure Santos Dumont. We based our optimization on the activities related to alignment, to read and statistics generations, and also about that, we do at the final 2 types of comparisons. How about the behavior in the SCCD architecture, and how about the lustre activity? It's some difference in performance, and we obtain the result. Yes, one can be improved if we also, after modeling of my workflow, my decision of which one is the architecture that's going to be used, is very, very important at the time of executing my workflow. And then all this optimization is because my modeling is like in evolution also. This is my 3 activities of my workflows. Also are all of them, both of them are executed in lustre, and we obtain the following result. If I introduce these 2 types of activities for split my data in here and for joining my data, for that, and use it moving also the version of the bow tie for threads, we can obtain the performance improvement of 70% between them, including the activities to that. Yeah, this is the first result that is related with the variability of my workflows in modeling. And this one other result here that there are two types of parallelizations. If I call the parallelization of my activity multi, I have a first level of parallelization, but if I call also my treats in my system, my workflow system, in this case parcel, I can obtain more gain of my total execution time here. Then after the double parallelization of my software and of my system, we can obtain better results in performance and are also more in performance than scalability. But here with 6 nodes is my limit that we can observe that are the best results here. Well, there are more uses here. For example, for one entry, also only for one entry, we can, if I use the Lustre, there are two types of architectures, Lustre or SCCD, we can approximately gain 2 or 3 minutes, but this is only for one execution. Imagine for several types of executions, hundreds or thousands executions that we need to do. Okay, I finished my presentation, I think, but I'm going to, okay, this is the next steps. The next steps is more related with the data analytics, because we needed to join some genomics data with the transcriptomic data, and we cross this type of data with the data that is generated. It is based on cardiomyocytes, but in here, this is other type of workflow. It's more analytic, that time and computational consuming, and this is other point of view that we need to explore more in modeling than in executing. Okay, this is other workflow. This is one of that is from genomics to how I'm going to use the different type of databases to in this case the workflows. The difference is maybe it's not automatic. Why? In that one activity is the inclusion of the human in the loop. Why? Because we can automatize here, for example, okay, I'm going to execute automatically the alignment and the processing A, B, C, or D, but in one time, the human need to resolve some problems. Okay, which one is the reference, or if the reference in the publication, for example, or if I need to include more data, or if I need to decide to exclude more data, etc. And then this is other type of workflows that are not based on execution and more based in modeling. But in some activities here, for example, that is related with the generation of clean data for genomics, here can be the sub workflows in this all workflow can be optimizing. Well, after that, these are my one of my present research. We are interested in also in developing science gateways, and then science gateways for web portals. But the thing is that today this workflow, this portal hosted different type of software and different type of workflows. When we finish the workflow and we needed to deploy to the system, and this is a very consuming and intensive time, because we need to test this type of architecture. But the thing is that one point that is very, very interesting here is, okay, when I have my architecture in for example, of the bioinfo portal, we have 4 levels. The level of the web interface, the database, the execution, and the hard resources, for example. But we want to start with a smart and intelligent gateways. And if we have the different type of executions, we store in my database the different executions of my different software, etc., we can apply machine learning to cross information about size of the data, about the number of the threads that are used for the execution, and for example, the number of bootstrap of the number of one parameter that need to be used. And then the idea is that I use my my metadata, my provenance history that is related with the experiment, with the biological data, and my computational results to predict which one is going to be, for example, for this type of experiment that I use transcriptomic workflow based on my previous results is more, for example, it's probably that using for 6 nodes in that architecture is going to be the best decisions for decrease my computational time. And then this is I have a master degree student for that, and my collaboration that are specialists in machine learning and HPC and me and bioinformatics, and then that is that we are developing at the moment. Well, as the conclusion is like bioinformatics and genomics and transcriptomics and all phylogenomics, that is my favorite, and all the omics analysis are a very nice case of study that can benefit from the features of the HPC environment. Obviously, have it's a multidisciplinary sport. I really have a little effort that, for example, okay, the bioinformatic decision, the biological decision, the architecture decision, but in clouds and in supercomputer, we obtained a very nice results, and we're happy about that. And then thank you. Well, thank you so much. This is my institute in Petropolis here in Rio de Janeiro. Thank you so much for the invitation, for my lab, for the Laboratory of Bioinformatics, my institutions, the CINAPAT and the Santos Dumont for the architecture and all the services. And if you have any questions, you have my contact, and you can, if there is any question, please.
Questions? Thank you, Carol, very much. Very good talk. Very good explanations. So I have one talk. I have one question. What about single cell RNA? Single cell RNA set. The workflow you have been talked about can be applied for that, or must be adapted?
Yeah, I think that is this is a case of study. I think that for in totally for RNA-seq or for different type of analysis of transcriptomics, I think the problem and the cases of study is very similar. Maybe changing the software, or maybe changing the database that are used, and the type of data that are included. But the thing is that my collaborator in Germany, the Laura, she's a specialist in single cell, and he, for example, it's like I need to direction, because my specialization is bioinformatics and workflows, and she told me, okay, Carrie, there is here, this is the specific data. We need to construct this type of database because there is not exist any type of database specifically for cardiomyocytes for single cell. But I think that the modeling is the first step for any problem, for any experiment. And but the thing, my point of view with that is like when we model, we're starting to model, we need to fix which one is the data, which one is that I'm going to use. If for single data set, for example, I'm going to use different type of parameter, etc. Then I think that with the evolution of the different omics analysis, I think that the problem is the same. Be careful with the data, with the software, with the infrastructure, and it's a multidisciplinary problem. Yeah, I'm not a specialist in single, but I'm supporting the bioinformatic modeling of a specialist of the single RNA-seq, and I think that that is my expertise. For example, the other thing is that she's not thinking about, okay, I'm not interested in the architecture, I'm interested in the data. Okay, but for that, for example, my experience with her, it was she was a protocol, but the first step that I did is, okay, I can do that automatically, and then after that I can move that with a supercomputer, and then this modeling, it can be done for my group. It's okay, but the expertise from the biological data for experimental data in weight, this is a very transitions that from biological data to bioinformatic and to HPC environment. But it's a multidisciplinary study. And it's nice for that. It's nice to interact with other groups.
Great. So, thank you very much, Karen, again.
Thank you.
And we are done for today, and we will be back tomorrow at 9 a.m. See you there.
Thank you.
Thank you.